biochemistry subject mainly deals with the intercellular reactions or some sort of intercellular reactions which are called metabolism and their uses in our body, right? But before plunging into metabolism, we must know what we are going to metabolize, and for that purpose, we need to know something about the chemistry, the structure, the structural features, etc., regarding all the biomolecules. So, the major biomolecule comes carbohydrate, protein, lipids, and nucleic acids. So, all these, whatever we get in our surrounding or inside our cell, they fall on either of these four. So, you have to know when you are dealing with some molecule, biomolecule, you have to know first that which component, which compartment it falls into. Whether it is carbohydrate, whether it is protein with carbohydrate, whether it is lipid with protein or lipid with carbohydrate, nucleic acid with carbohydrate or nucleic acid with protein, etc., etc. So, the chemistry part is very, very important in order to understand the nature of the biomolecule, what we are dealing with. To begin with the chemistry of carbohydrate, we must first know the definition of carbohydrate. Now, may I ask you, anyone to answer whatever you know about carbohydrate definition? Anyone? Whatever you know about carbohydrate definition, anyone? Quickly. Slides are there. We can go, but first we have to inculcate about our previous knowledge, existing knowledge. We must form the base, then we can form the building. So, a pre-existing knowledge is very, very important to inculcate. Yes, anyone? There? Why don't you sit in front? Is something wrong in these seats? Come. Both of you. Come. These are your first class. From the very first class, if you are going away from the teachers. Why? Yes. Carbohydrates are chains of sugar molecules. Fine. Anything else? Aldehyde and ketone derivative of alcohol. Optically active polycarbonate. Carbonyl compound. Polycarbonyl compound. May I ask you something? What do you mean by polycarbonyl? Mistake हो गया, है ना? Glucose has only one carbonyl group. See now, fructose 2. Yes. Polyhydroxy. Yeah, that's better. That's better. Anything else? Anything else? Anything else? Anyone? Yes? Derivative. Omit this word derivative. It is not derivative. Derivative means something which is derived from. So you are not changing polyhydroxy compounds. These compounds are unique, they are independent. Correct? Do not add derivative everywhere. Why? That is a general formula, empirical formula. That formula might coincide with many other compounds which are not carbohydrate. So basically, what is your name? Tathagata. What Tathagata said was nearest to the perfect definition of carbohydrate. Now we'll go to the definition of carbohydrate. Now take half a minute, 30 seconds to read carefully each line and each word of this definition. Yes. Now someone please take the pain to make me understand or explain the first and the second part of the definition. What Tathagata said resembles 99% with the first part. Do you agree? What about the second part? Carbohydrates are polyhydroxyaldehydes or ketones or আমার ঠিক আছে 50% ঠিক 50% ভুল এস্টার না গ্লাইকোসাইডিক লিঙ্কেজ ইথার লিঙ্কেজ ঠিক আছে ইথার লিঙ্কেজ কখন হচ্ছে দুটো অ্যালকোহলের মধ্যে হয় আর যখন একটা অ্যালকোহল একটা কার্বনিক গ্রুপ হয় তখন তাকে বলে glycosidic. So the first part make us understand the definition of glucose, fructose, mannose, xylose, ribose, ribulose, xylulose, etc., etc., etc. But when it comes to lactose, maltose, isomaltose, trehalose, cellobiose, cellulose, starch, amylose, amylopectin, etc., etc., inulin, heparin, heparan sulfate, creatine sulfate, damotan sulfate, then the first part of the definition fails to give us the idea. For that purpose, the second one means if it is more than one monosaccharide unit, sugar unit, then the second part comes into rescue. Suppose you forget this second part to write in the examination, we will forgive. But it is better to know the exact one. So this is the best so far found definition for carbohydrate. Carbohydrates are polyhydroxyaldehydes or ketones or the compound which produce the same. Same means polyhydroxyaldehyde or ketone on hydrolysis. Fine. So that encompasses not only monosaccharides but also disaccharide, oligosaccharide, polysaccharide, everything. Fine. Even the glycoproteins. Glycoproteins. Now, how do you classify carbohydrate? The olden classification was only mono, oligo, and poly. But then, slowly, disaccharide units were found very, very important. So, many disaccharides were involved, were discovered, and their uses also was discovered. So, they made disaccharide a separate class because of the importance. That's all. Fine. So, now the present classification is 4. Mono, di, oligo, and poly. Simple. Understanding is very simple. Mono means having one sugar unit or saccharide unit. Use the word saccharide. Saccharide unit. Fine. Saccharide unit means what is a monosaccharide? Which cannot be hydrolyzed to form a simpler, means simple than the other one. See, a simpler carbohydrate. তার থেকে সরলতর অন্য কোনো কার্বোহাইড্রেট তৈরি করা যাবে না। এমন একটা সুগার ইউনিটকে বলা হয় মনোস্যাকারাইড বা স্যাকারাইড ইউনিট। ঠিক আছে, যেমন জাইলোজ। জাইলোজকে ভাঙলে আমরা যেটা পাবো that is no more than that is no more carbohydrate. Right? So similarly, আমরা যদি ল্যাকটোজকে ভাঙি তাহলে গ্লুকোজ প্লাস গ্যালাক্টোজ পাবো, তাই না? So lactose is a carbohydrate. Now disaccharide, where two monosaccharide units are covalently linked. What do you call the linkage? Glycosidic linkage. Always remember. Carbohydrate, the carbonyl group forms a linkage. It is called glycosidic linkage. Clear? Very, very important. There are many glycosylation enzymes whose deficiency causes many diseases which you are going to treat. Next, oligosaccharides. When the number of sugar units is between 3 to 10, above 2, below 10, up to 10, 3 to 10. So trisaccharide, disaccharide, pentasaccharides. So up to pentasaccharide, we have to remember at least one example. Fine? We'll come to that. Polysaccharide, more than 10 monosaccharide units. More than 10 monosaccharide units. Fine? Now, how do you classify monosaccharide? We can classify on the basis of their functional group. Classification of monosaccharides on the basis of their functional group. First, they might contain aldehyde or keto group. Both are called together carbonyl group, but they are different, aldehyde and ketone. Aldehyde group remains in the terminal carbon, keto group cannot remain on terminal carbon, at least one carbon away from the terminal carbon. কারো যদি বুঝতে কোথাও অসুবিধা হয়, please raise your hand, because initially it is very easy to remember. So don't hesitate. So for that we have two classes, aldoses which contain aldehyde as functional group, ketoses which contains keto group as functional group. Examples are given. It is you who have to remember and write in the examination. Sometimes the examiner can ask, name 5 aldoses, name 5 ketoses. Simple question. If I remove the slide now, can you name, only 3 are given. Can you name the other 2? Is there anyone who can name other 2 ketoses? See? Apparently, humble looking question becomes very deadly. If I ask you to name 20 enzymes, can you name? So if I point it out, you will be seeing that you know more than 20 enzymes, but you don't know that you know that. Similarly, you must know that what you don't know. First year is very simple thing. You have to know what is what, that's all. Definition and what is what. This is this, this is this, this is this, this is this. Interconnection and everything will come later. Only thing is you must know whatever, so that from the next year onward, whatever compound is named on the board, you should be knowing that which type of biomolecule is that. It's a glycoside, or it's a lipid, it's a glycoprotein, glycolipid, phospholipid, sulfolipid, whatever it is. This much, that's all. This is all about first year biochemistry. Basics. That's what it's called basic science. Now, glucose, galactose, mannose. Can you name any other aldose sugar? Fructose. That. Take a wall. Glyceraldehyde, very good. Very good. Now, may I ask you to take the pen to draw the structure of these 2 compounds. Most used 2 monosaccharides, glucose and fructose. First you take the pen to draw the structure quickly. Then I will explain something. Yes. You have to draw the structure like this. So how many carbons are there? Both are having 6 carbons. Both are having 1 carbonyl group and 5 hydroxy group. The 6th carbon, see, the 5th and 6th are same. Same. First and second you cannot compare because there are 2 different groups. First one is having aldehyde group and second one is having carbonyl keto group on the second carbon, because keto group cannot be on terminal carbon. It cannot be on 1 or 6. Now, I think you should not have any doubt why I am calling the aldehyde group carbon is 1. Is there anybody who is having doubt? I could call this 6th, C6, but which one is correct? the aldehyde group carbon is C1 or C6? I think you know that reason. What is the reason? Yes, more electronegative. More electronegative, that's why it comes first. It's called polyhydroxyaldehyde, not aldehyde alcohol, but alcoholic aldehyde. Right? Fine. So this carbon becomes C2 then by the same rule. Correct? C1 and C2. They are not different, they cannot be compared for any difference because they are two different functional groups. But what about third carbon? Are they same? Yes, they are also same. So in the glucose and fructose, everything is same in the structure except the functional group. Is it easy to remember? Only the third carbon is having OH group on the left hand side. Only the third carbon is having OH group on the left hand side. And rest all are same. Fine? Remember? Memorize the structure. We'll come to that later, that series. Now, we can also classify monosaccharides on the basis of the number of carbon atoms. Once, an external examiner asked this question to 20 successive students. None of them could answer. None of them could answer. See, it's apparently humble looking, simple questions, so deadly. If you don't know. So what are those? Triose, tetrose, pentose, hexose. Means 3 carbon, 4 carbon, 5 carbon, 6 carbon, 7 carbon, 8 carbon, 9 carbon and 10 carbon. One example also given. Up to here glyceraldehyde, erythrose, ribose, glucose. But for after that, glucoheptose, just add gluco prefix. If you don't remember, add gluco prefix. That's all. Heptose. See? Isn't it easy? Huh? One Viva question answered. Sometimes you ask that after theory examination, sir, what are the important things to study for Viva? Whatever was important for your year has already been given in the theory paper. Still you are asking, तुम्हारा नाम क्या है बसंती? है ना? Huh? Whatever was important for your year is already given in the theory paper. So what will you study for Viva? The theory paper, which is already out. So simple. Huh? Then disaccharide. Coming to disaccharide having more than two monosaccharide units, and they can be divided into two classes, which is based on their reaction with certain reagents, say Benedict reagent, whether they have reducing property or not. Why they are having this classification? Because the glycosidic bond might involve the carbonyl groups. So in that case, there will be no free reducing end for the disaccharide. So they cannot reduce anything. So they can be either reducing or non-reducing. So what are the examples of that reducing and non-reducing? Reducing examples say lactose, maltose, isomaltose. Write down. Lactose, maltose, isomaltose, cellobiose, cellopen, CELLOBIOAC. Cellobiose, correct? So these are all reducing disaccharide, non-reducing disaccharide. Can you name anyone? Yes. Very good. What is your name, Baba? Huh? Garibal. Jagdeep. Jyotirmoy. Mahapatro. You are from? Which district of Odisha? Bangla ball. Half the people speak Bangla over there. Huh? Bola sir? তাই তো? Half the community is Bengali. They marry Bengali girls. Anyway, good. Very good example. Free halos. Glucose plus glucose. C1 glycosidic linkage. Okay? The tougher one, he answered. I want the easier one. Example. Sucrose, the table sugar. Correct? Fine. One next slide. The structures. We'll come to that later. This is the structure of sucrose. This is the structure of sucrose. Here I want to explain something. In your books, is there anyone who has any question about the way the structure have been shown here? What is it called? What do you call this kind of structures? Bolo. Pyranose and pyranose ring structure. Very good. Very good. Very good. So, let me tell you the features of this kind of structure. This structure actually shows 1, 2, 3, 4, 5, 6. A 6-membered heterocyclic ring, pyranose ring. A 5-membered heterocyclic ring, furanose ring. Now, the features is these all 5 carbons are in the same plane. Actually, they are not, but when you draw the structure like this, you maintain the feature. This is a way of expressing a 3-dimensional structure on 2 dimensions. We live in a 3-dimensional world. We can express in 2 dimensions. If you go to some extraterrestrial planet where the normal dimension is 4D, so there children could make 3D diagram. Here we cannot make. So there is always -1 dimension we can express. -1 dimension we can express. We live in 3-dimensional world. So we can express in 2 dimensions. So now the molecule we are showing, it is like a plane. So where in the plane, all these 6 member or 5 member in the same plane. The OH which is jutted out upward, it is above the plane like this mic. The OH which is below the plane or like this. Right? This shows, depicts that it is whether it is above the plane or below the plane. And rest all are in the same plane. Fine. But actually it is not. And for that we need to understand Haworth projection. Haworth projection. This is Fisher's way. Emil Fisher way to express 3-dimensional structure in 2 dimensions. Fisher projection this is. But actually they exist in Haworth projection. Fine. So now what is important here in the structure is, in many books you will find the structure of sucrose where this glucose is drawn this side or side, and this OH has changed their direction. The OH which is shown here upward, it will come downward. The OH which is downward, it will come upward in book. And you will come with the book to us, to me, many times they came. Sir, whatever structure you wrote in the book, in the board, is not matching with that in the book. Who is correct? I said both. When you put the glucose molecule above this fructose molecule, you can maintain the direction like this. The 3rd carbon is having OH on left hand side. See the structure you wrote? Glucose and fructose. If the OH is on the left hand side of a open chain form structure, this is pyranose form structure. The open chain form, whatever OH is on left hand side, they go below the plane. Above the plane. in the right hand side, they go below the plane. Previously we used to remember, now we cannot say that way of remembering. Previously, our state was ruled by a leftist, Bampunti. So say beta form means the left hand side rules and alpha goes down. Correct? Now we cannot say that example. But in the back of mind you can remember like this. The beta goes up, alpha goes down. Huh? Like that. So anyway, politically it will not be suitable to use the example anymore. Now, so like this, so if it is left hand side, it goes upside, above the plane. If it is right hand side, it goes below the plane. Now see, what is there? The third OH of glucose molecule is OH above the plane. Fine? This is 1, 2, 3. Third OH above the plane. Right? When the same structure in your book is drawn, it is not drawn like this. Rather, this glucose comes side, this side. So this one, this group will come like this. Like this it will become. Okay? So then, in order to maintain the structure like this, make this bond folded like this, for drawing purpose in the book only, they shift the change here. This OH below and this OH up, as if the molecule has flipped it, so that we can fold the glycosidic bond upside down. Followed? This is the only difference in the book and this diagram. Not mention what they have done, I have to mention what I am doing. Clear? We come to oligosaccharide quickly. There are two types of oligosaccharide, O-linked and N-linked. O-linked oligosaccharide and N-linked oligosaccharide. Oligosaccharides are mostly found in association with proteins, and these compounds are called glycoproteins. Proteins associated with sugars, carbohydrates, are called glycoproteins. So oligosaccharides are mostly found in association with proteins. The proteins, there are many amino acids. Proteins are made up of amino acids. So some amino acids contain functional OH group on the R group, extra group, and some protein contain extra amino group. So the OH group of hydroxyl group, oxygen of hydroxyl group, if attached with the sugar unit, then it is called O-linked. And if the nitrogen group of amino group, extra amino group of amino acid, is attached with the sugar unit, it is called N-linked. Fine? So there are examples like amino acid which contain OH group, serine and threonine, usually found in acid with O-linked glycosidic, and arginine, histidine, all these are found, mostly arginine are found, lysine, they are found in acid with N-linked glycoproteins. What are their functions? O-linked glycosides have the roles in recognition, interaction, and enzyme regulation. Can you remember the cell surface receptors and all everything? They are all glycoproteins. Most of the proteins in our blood, serum, are glycoproteins. Right? But but fallacy is, most of the proteins in the plasma are glycoproteins, except albumin. Human serum albumin is not a glycoprotein. It is a pure protein. Okay? Simple protein. Unlike bovine serum albumin or sheep serum albumin, whatever it is. Next, N-linked. Okay. So these are important in the sense when you have to study the blood group antigens, you have to remember everything. Then it will be easier for you. At least remember what is what. There are oligosaccharides which can be divided into groups, O-linked and N-linked. O-linked are synthesized on site in situ on site or in situ. N-linked are like making the frame outside and then coming and joining with the structure. Followed? Like these, the oligosaccharide chains are synthesized. This is one of the difference. Only synthesized in situ, N-linked are synthesized and then got to connect with the compounds. Okay. Some example of this. See the oligosaccharide chain. N-acetyl nuraminic acid, N-acetyl nuraminic acid, N-acetyl nuraminic acid. Gal means galactose, N-acetyl glucosamine, NAG, N-acetyl glucosamine, mannose, N-acetyl glucosamine, N-acetyl. This is, if you see, fucose, ASN, asparagine, asparagine, glutamine, lysine, they all contain extra amino group. Correct? Asparagine, glutamine, these are all amino acids. They are amide containing amino acids. Okay, FUC means fucose. These are all blood group antigens. ABO, blood group antigens, they are all glycoproteins. Bolo, fucose, like a sugar, extra sugar. Polysaccharides, quickly. Polysaccharides can be of two types, homopolysaccharides and heteropolysaccharides. Homopolysaccharides where all the participating glucose units are identical. Heteropolysaccharides where all the monosaccharide units are non-identical. Or a repetition of disaccharide unit. Okay? These are same, all are not same, non-identical. Right? All are same, identical, homopolysaccharide. Example, starch, glycogen, inulin. Heteropolysaccharide, plenty are there. Plenty are there. Hemicellulose, lignin, pectin, sibirine. And then mucopolysaccharides, heparan sulfate, heparin, dermatan sulfate, creatine sulfate, conotine sulfate, etc., etc. So already you know about something called dextran, agar, cellulose, the linkages, hemicellulose, heparin. Any question upon this? Starch, storage form of carbohydrate in plants. Partially digested starch form dextrin. Glycogen, storage form of carbohydrate in animals. Inulin, polyfructosan, dextran, galactose, glucose, dextran, glucose, but linkage is different. It is not alpha 1,4 or 1,6. Agar, D-galactose, polymer. Cellulose, beta D-glucose polymer. Glycogen is alpha D-glucose polymer. This is beta D-glucose polymer. That's why we cannot digest cellulose. We cannot digest. সব রকম পুঁই শাক, পালং শাক, ঝিঙে, কুমড়ো, আলু বাদ দাও। হ্যাঁ, সবজির নাম করো। নোটে শাক, বাতুয়া শাক, ইত্যাদি 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 যা আছে শাকসবজি। cellulose. We can eat, but we cannot digest. It's called dietary fiber. So this is beta 1,4 glycosidic linkage. But whereas glycogen, do you eat glycogen? Is there anyone who has taken glycogen ever? তুমি গ্লাইকোজেন খেয়েছি খাওনি বলো, তুমি গ্লাইকোজেন খাও রোজ? গ্লাইকোজেন খাও কি খাও না বলো? শিওর? তুমি আজ পর্যন্ত চিকেন, মাটন, মাছ খাওনি? গ্লাইকোজেন স্টোর থাকে কোথায়? লিভারে, মাসেলে, সেটা তো পেটেই যাবে আমাদের, তাই তো? তারপর তো ডাইজেশন হবে, তাই না? So see, so interesting, huh? Carbohydrate, it not only gives us energy but also protects our body from here to here, all carbohydrate. Huh? Carbohydrate. Not the polyester one. The cotton one is carbohydrate. Fine. Inulin, other cellulose, hemicellulose, heparin, heparan sulfate, suberin, etc., etc. Next. Isomerism. So first question comes from carbohydrates. What define carbohydrate? Classify carbohydrate. First question done. Second question is isomerism. Describe the different isomerism or different isomers of carbohydrate. Right? It's a very long question, but we will learn how to make it short and concise for our examination purpose. Isomerism. Do not write. Do not write. This is for only +2 chemistry. You have passed +2 chemistry, so you need not to write anymore. What is there? Structural isomer and stereoisomer. There are two types of isomerism. These are the structural isomerism, chain isomerism, positional isomerism, functional isomerism. We will take few of these, few of those. Stereoisomerism, geometric isomerism, and optical isomerism. So you should be able to know that you will tell what is optical isomerism and geometric isomerism. What is that? For your what? Revision purpose, this slide is made. So that you don't ask me further what is geometric isomerism. After I remove this slide, move on to next slide, I will know that you know what is called geometric isomerism. I will know that you know what is called stereoisomerism. I will know what you call positional isomerism. So just go through quickly about the important points before I move to next slide. And I will not teach this slide. Why? This is in class 2. You are in first year. Fine? Done? All of you? Don't look at me, Baba. I am not so handsome. Look at the slide. Lend me your ears, not eyes. This is a lecture class. Fine? Done? Geometric isomerism? Now, under the heading of isomerism of carbohydrate, we will study these. At least these 5. Fine? So we will first, what we will do? We will explain each one of them by definition and give one example. That's all. Very simple. Very easy to score marks. Good marks you will get. And the student who will be able to draw the structure, he will get very good marks. But I tell you, drawing structure does not carry any extra credit unless the question is asked to draw the structure. Am I clear? Suppose you are asked to describe a metabolic pathway, and if you draw with the structure, by the rule of the examination, the one who has drawn the structure and one who has not will be same. Will be evaluated as same, identical. There is no extra credit to be given to those who are drawing the structure, right? Unless it is specifically asked, like draw the structure of cholesterol. There if you don't draw the structure, you lose marks. But describe glycolysis, if you don't draw the structure, you will not lose marks. You have to mention the names, the enzymes, the coenzymes, etc., etc. But when you draw the structure, there are some students who draw. Every year, there are 99% students who never draw, but 1 to 2% students, they will draw. Beautifully they will draw. They will explain everything. we keep some marks, if not in that question, for some other question, we will give that. Correct? After all, we also human, we also have human heart. If I like someone's answer and I cannot give more than 5 marks because the highest marks is 5, so I will look into that answer paper where he has not been so much, not so good in some other question, I will give 2 extra marks there. Followed? We are human beings, we can do that. So that's why. Is it clear? By rule. You must know the rule, that's all. D and L, capital D and capital L. Next one is small d or positive or small l or negative. Next one is anomalism. Next one is epimerism. Next one is pyranose and pyranose forms. Is there any other form? Yes, one more thing here I have not mentioned. These 5 should be sufficient for your level. D and L configuration. Very easy to understand. It depends on the OH, the location of the OH on penultimate carbon. Last but one carbon. Last এর আগের কার্বন। লাস্টের আগের কার্বনে OH যদি ডান দিকে থাকে তাহলে D, বামে থাকলে L। Fisher projection। কোথায়? In Fisher projection, if the OH in the penultimate carbon atom resides on right hand side, we call it D isoform. This has nothing to do with dextro. This is only D. D means D, that's all. That is the beginning, that is the end. D stands for nothing else. It is not a short form of anything. কি বলবি? এই বলবি তোরা? এই বলার? বলার? কি বলবি? So D and L. L is where the OH is on left hand side. Now, what is the smallest carbohydrate? Smallest aldose, D-glyceraldehyde. Smallest ketose, dihydroxyacetone. If you go below that, means there should be at least 3 carbon. Why? The definition says polyhydroxy. Poly means at least more than 1. So it must contain at least 2 hydroxy group and then 1 aldehyde group or keto group. So at least 3 carbon should be there. Below 3 carbon, it is not carbohydrate anymore. This is D-glyceraldehyde. And what is this? Is there any compound like that? How many of you think there is a compound like that? Please raise your hand. Let me take a poll, opinion poll. How many of you think there is a compound like this? Please raise your hand, quickly. Quickly, choose yes or no. No one thinks that there is a compound like this. Please raise your hand, yes, louder. 1, excluding me. I cannot be counted, na? 1, 2. Only 2 of you think there is a compound like this. Right? Yes, there is a compound like this, but that is not carbohydrate. Now, only 2% have answered there is a compound like this. They have to name the compound. You only said it is existing, you should name it. IUPAC name to বললি হবে না কেন ওটা বাদ দাও অ্যাকচুয়াল নাম বলো আমি লিখেছি তো বলি হ্যাঁ জানতাম oxalic CHO bond CHO. হয় কি? CHO bond CHO কিন্তু আমি এখানে দেখি দুটো কার্বোহাইড্রেট দুটো করতে হবে, তাই ভালো করে দেখিস, খেয়াল করিস। Oxalic acid. Now, this is no more ভুলি গেল নাকি? হতে পারে memory can sometimes replace each other. হ্যাঁ, totally it can মানে misguide you. দেখছি আমি। Now this one, can anyone see from distance? Now this is a series of aldoses. From here to here, each step, the number of carbon atom increases by 1, and each time the newly added carbon occupies the second position. So second position can have OH either on left hand side or right hand side. Now this is by convention. When you draw the isomer on the left hand side, you draw the OH of the newly added carbon. When you draw the isomer of on left hand side, the OH of newly added carbon atom should be on right hand side, and for right hand side vice versa. So the newly added carbon is this one. So rest all these two are same, but this one will having for left hand side if you are drawing, OH will be on right hand side. For right hand side if you are drawing, OH will be on left hand side. The newly added carbon OH. This is newly added carbon for aldoses. For aldoses C2 is a newly added carbon. For aldoses C2 is a newly added carbon. For ketoses C3 is a newly added carbon. Good. So now this one is D-glyceraldehyde. See the plus sign. You see here all are D, but some are plus, some are minus. Okay. So that D and small d, big D and small d are different. So this is erythrose, this is triose. So how do you name? ET, erythrose, triose. Then ribose, arabinose, xylose. How do you pronounce it? Anyone quickly? Lysose. X-zo. Lysose. L-Y-X-O-S. Now coming to this is for pentose, right? This is for 4 carbon. This is 5 carbon. What about 6 carbon? Can we remember? Yes, we can remember. If we remember the order, we can draw any one structure. All we have to remember is 2 things. The newly added carbon for aldoses C2 and OH will be on left hand side if we are drawing the structure on right hand side. OH will be on right hand side if we are drawing the structure on the left hand side. That's all. These 2 lines. Then we can extend it up to any number of carbon atoms and we can draw the structure. Even the unknown carbohydrates, they were long described by their structure even before they were actually discovered. alchemists gladly make gum in gallon tank. All aloes, alchemists, altros, gladly, glucose, make, mannose, gum, glucose, in, iodose. Then gallon, galactose, tanks, talose. How to name them? You know the mnemonics for remembering the name. If you remember the names, the order, you can draw the structure. So drawing the structure of glucose, fructose, now you need not to memorize it. You can draw it yourself. Only thing that you remember, the first initial structure, diglyceride, and two rules, golden rules. That's all. So we will stop here, and next week, tomorrow there is no classes probably. Tomorrow 9 o'clock there is no class. You'll have some rally. You have to walk. Morning walk. Okay? And next week you'll have class, then 2 weeks probably, I don't know. We have semester examination. So we'll be busy for the first week of January. Second week we are trying to conduct the classes, at least the theory classes, during the Pushbanga Mela. We'll try to conduct the theory classes if possible. Because we'll be losing many weeks then. 2 weeks. Long time. Become very serious. No time. Now this one, your course will be for 13 months. So just remember which month you joined. Which month? So, next year, December, you will have exam. 13 months, 2nd November, 13 months. Exam. Many of you are not yet admitted. Half the, 2/3 of the class is yet to come. What they will do? Time pressure.
Understanding of tissue elements. Since which class? কোন ক্লাস থেকে আমরা টিস্যুস পড়ছি? কোন ক্লাস থেকে আমরা অ্যানিমেল এবং প্ল্যান্ট টিস্যুস পড়ছি? হ্যাঁ? 7-8 তো? হ্যাঁ? আমাদের আন্ডারস্ট্যান্ডিং কতদূর? What are the types of intercellular filaments? Fibrous structure, intercellular fibrous structure কি আছে? একজন। ওই ছোটবেলায় যেরকম কান ধরে মুরগি করে রেখে দিত, হ্যাঁ ওই রকম করে রেখে দেব। কোলাজেন ইজ এক্সট্রাসেলুলার ফাইবারস। কোলাজেন ইজ এক্সট্রাসেলুলার ফাইবারস। ঠিক আছে? somebody else, বসো বাবা, আমি রাগ করি না, মুখে যাই বলি আমি, ঠিক আছে? এক্সট্রাসেলুলার ফাইবারস, ইন্ট্রাসেলুলার ফাইবারস কি আছে? কিরে, এখন তো 3/4 থেকে টিস্যু হিস্টোলজি প্লান্ট, হ্যাঁ ইয়েস অফকোর্স। মাইক্রোফিলামেন্ট, হ্যাঁ ইয়েস, one type of intracellular fibers is microfilament. Thank you. Sit down. The others are microtubules and intermediate filaments. Others are microtubules and the intermediate filaments. ঠিক আছে? Being a student of medical graduate, you must know and remember each and every part. আমাকে মেমোরাইজ করতেই হবে, এই জিনিসটা মাথার মধ্যে ঢুকিয়ে ফেলতে হবে। ঠিক আছে? Microfilaments are 5 nanometer in diameter. Write down. Put a heading. Intracellular fibrillar structure or fibrous structures. Number one, microfilaments which are of 5 nanometer in diameter. They form the cell cortex, that is below the membrane. ठीक है? You need a fibrous network to maintain the shape of a cell, isn't it? At a cell membrane রইলো without any support, collapse করে যাবে. So there is a submembranous network of microfilaments which are of 5 nanometer in diameter. The next is the microtubules. You know something about microtubules as I can expect from you. That is the microtubules in the flagella, in the cilia, and in the dividing cells organized from the centrioles. That 9+2 organization. That 9+2 organization. Don't you remember this? এরকম তিনটে কিসের স্ট্রাকচার ছিল রে? কার স্ট্রাকচার ছিল এরকম? This is a basic structure for cilia, flagella and the microtubules in dividing cells from the centrioles. তাই না? ঠিক আছে? So these microtubules are formed out of tubulin proteins. There are 11 chains in the wall of a 13 chains in the wall of a microtubule. ঠিক আছে? Having a diameter of 25 nanometer. So 5 nanometer is microfilaments, 25 nanometer is for the microtubules, and there are intermediate filaments which are of 10 nanometer diameter. ঠিক আছে? Microvilli in the tall columnar cells of intestinal epithelium, ঠিক আছে? Those are endowed with the property of secretion as well as absorptions. You know the microvilli? Tall columnar cells এর এই basement membrane এরকম একটা tall columnar cell, এইটা nuclei, this is the nuclei, ঠিক আছে? And at the surface there is hair-like projections. These are the microvilli. You know this. I know you know this. All right. These structures are again supported by the microfilaments. These structures are again supported by the microfilament. So these are the fibrous structures which are found within the cell. That is, these are intracellular. And outside the cell, that is extracellularly, we find mainly the collagen fibers and the elastic fibers. Nowadays, reticular fibers are considered a type of collagen fiber. ঠিক আছে? You can call it that extracellularly we find collagen fibers, reticular fibers and elastic fibers, whereupon you should write that nowadays reticular fibers is a subtype of collagen fibers. Now, how does a zygote grow into a full embryo? full fetus capable of individual life, there are only two processes, that is the growth and the differentiation, isn't it? The single cell zygote will become a full blown newborn baby out of two processes. Number one is the cell division or the growth and the cell differentiation, isn't it? So in growth potential, these are expressed in number one, mitotic divisions, else in other sites, especially in the germ cells, we see the meiotic division. So the division or the multiplicative growth. Another is the accumulation of intracellular substances, that is the auxetic growth, and there is accumulation of intercellular substance, that is the accretionary growth. All right, these three kinds of growth is possible, which is multiplicative, which is auxetic, and another is accretionary. In multiplicative growth, we see we can classify body cells into three categories. Number one, after birth, some cells do not even divide to the slightest amount, that is the neurons. So these are permanent cells. They can't divide after birth. All right. Number two is the proliferating cells, which has a rapid turnover because they need to maintain a static number of cells. These are the intestinal epithelial cells, our skin cells, and the myeloid precursors of RBC, WBC, platelets, etc. All right. So these, due to wear and tear, they lose at a constant rate and they must be replenished from the stem cells. So this is the category 2. And the third category, there is another category, that is the hepatocytes. Though they are capable of dividing, but they remain quiescent until and unless 2/3 of the liver cells are removed. So they remain as quiescent cells. They remain as quiescent cells. You see another variety, that cell division starts when there is an imbalancing nucleocytoplasmic ratio. You have heard the term nucleocytoplasmic ratio, that is the cross-sectional area, cross-sectional nuclear area divided by the cytoplasmic area, which is normally 3:1 to 4:1, which is normally 3:1 to 4:1 in erythroblast, in myeloblast, you can see that this ratio is maintained. When these cells differentiates into, suppose for platelets, megakaryoblast will give rise to platelets, the erythroblast will give rise to RBCs, the lymphoblast will give rise to lymphocytes. When these sites appear, the ratio becomes 2:1 to 1:1. So their dividing potential, we can identify that their dividing potential has been reduced, isn't it? What happens in the developing oocyte? What happens in a developing oocyte? The cell size increases by auxetic growth. Cell size increases by auxetic growth. That is, they accumulate intercellular substances. All right. And for a large cell, surrounding cells are unable to provide nutrition to such a cell. And numerous flattened cells appear around it. Numerous flattened cells appear around it, which are known as the follicular cells, which gives nutrition to these developing cells. তোমরা পড়েছো? I know তোমরা পড়েছো জিনিসটা, ঠিক আছে? এরকম একটা ওভাম যখন গ্রো করছে, ভেতরের লেয়ারটা ওভামের সাইটোপ্লাজমিক মেমব্রেন, বাইরেরটা হচ্ছে জোনা পেলুসিডা, ডাবল লেয়ার স্ট্রাইটেড মেমব্রেন, দ্যাট ইজ দ্য জোনা পেলুসিডা, ঠিক আছে? এটা কি থাকবে? Striated membrane, that is zona pellucida. This is a striated membrane. This is the nucleus. At present, there is nothing. Gradually, there will appear some surrounding cells, surrounding flattened cells from the tunica intima of ovary. These cells will provide nutrition to the developing or growing oocyte, and this will mature. Inside there will be the follicular cells that will give cumulus oophorous formation and the antrum folliculi or the মাঝখানে কি থাকে? একটা antrum folliculi থাকে না? ঠিক আছে? Blah blah blah. So this kind of cells produce the oxidative growth due to accumulation of intercellular substances provided by the surrounding cells. This is also seen in the large neurons. This is also seen in the large neurons surrounded by the neuroglial cells. So oxidative growth can be seen in developing oocyte as well as in the large neurons which are surrounded by the glial cells. The next part is the differentiation. Next part is the differentiation. From the zygote we see the, after the cleavage division, we see there are 4 cell stage, 6, 8, 12 to 16 cell stage, at which stage we call it a morula. And thereafter, after the formation is a space around the embryoblast, we will get the blastocyst. Alright. In this course, up to the stage of 8 cell stage, the cells are so much endowed with so much potentials that they can divide into a full embryo along with the membrane. So the cells of the morula up to the 8 cell stage can give rise to a full embryo with the membranes, means amnion and chorion. Full fetus plus amnion and chorion. Up to this 8 cell stage, the dividing potentials of the cells are called totipotent. These are called totipotent. So the totipotent character is maintained up to 8 cell stage. 8 cell stage of cleavage division. After this, these cells will not remain as totipotent. This means the next stage will be followed by the pluripotent stage, a plastic stage of pluripotent potential. Means this cell line can give rise to all type of tissues from the 3 germ layers. 3টে germ layer যে কোনো cell, যে কোনো tissue ও তৈরি করতে পারবে. But ordinarily they produce a particular germ line. Ordinarily তারা কি করবে? একটা যে কোনো either ectoderm, endoderm or mesoderm, যে কোনো একটা laminar যে সমস্ত structure ও দেবে. But by potential it can give rise to cells or tissues of any of the 3 germ layers. বোঝা গেল কথাটা? আমার potential রয়েছে, আমি এই পুরো বাড়িটা বানাতে পারবো। আমার potential রয়েছে। ঠিক আছে? On the 3 germ layers যে কোনো structure আসবে আমি বানিয়ে দেব। But ordinarily I am endowed to either I will form the ectodermal structure or the mesodermal structure or the endodermal structure. This is called pluripotency. Hello? So first totipotency, next the pluripotency. Alright. After this, there is a stage of chemodifferentiation. A stage of chemodifferentiation where due to some liberation of some chemical organizer material, we see there is appearance of primitive stick which acts as a primary organization. From the primitive stick develops the mesoderm and the notochord. The notochord acts as the secondary organizer. After that, from the notochord, due to its organizing influence, they differentiates the neural tube formation, which acts as a tertiary organizer. Which acts as a tertiary organizer by evocation and individuation, that is liberating of some chemicals and sending some cells or recruiting a cohort of cells into a particular line of differentiation. বোঝা গেল? মানে কিছু কেমিক্যাল লিবারেট করবে, যেগুলো কি করবে, কতগুলো একটা গ্রুপ অফ কোহর্ট অফ গ্রুপ অফ সেলস, ঠিক আছে, তাকে একটা ইন্ডিভিজুয়েশন মানে একটা পার্টিকুলার ক্যারেক্টার অ্যাজিউম করতে হেল্প করবে। দ্যাট ইজ দ্য ইভোকেশন অ্যান্ড দ্য individuation. After the chemodifferentiation comes the histodifferentiation. This means anatomically we will be able to find these cells is of epithelial character, these cells is of muscle tissue character, these cells is of neural tissue character. এই যে anatomic structures গুলো ওর মধ্যে এসে যাবে, shape, size and materials, ঠিক আছে, this is known as the histodifferentiation. After the histogenesis, we see the molding or organization. Groups of cells will be migrated at different sites. The entire space available to the developing embryo will be mapped into individuation sites. Means আমার এই পুরো বডিটা কেউ যেন এঁকে দেবে, ঠিক আছে, এঁকে দেবার পরে সে দাগ দিয়ে দেবে, এইখানে ভেইনস থাকবে, এইখানে লিভার থাকবে, এইখানে স্প্লিন থাকবে, এইখানে আর্টারিটা থাকবে, এরকম করে, ঠিক আছে, ম্যাপিং হয়ে যাবে। আর ঠিক সেই জায়গায় সেলসগুলো মাইগ্রেট করবে। সো দ্য মাইগ্রেশন অ্যান্ড দ্য মোল্ডিং, মাইগ্রেশন অ্যান্ড দ্য মোল্ডিং, ডিফারেন্ট টিস্যু শেপ। লিভারটা তো আর কিডনির মতো হয় না, কিডনি ডাজন্ট টেক দ্য শেপ অফ অ্যান আর্টারি। All right. This is the individuation state where the tissue characters are achieved, and the final modification is achieved through functional movements. যখন ফাংশন করতে শুরু করে তখন যে ফাইনাল মডিফিকেশনস হয়, ঠিক আছে, আর্টেরিওলার ডায়ামিটার, কোনো একটা ইন্ডিভিজুয়াল চ্যানেলের ডায়ামিটার, ঠিক আছে, তার ওয়াল থিকনেস, তার রেজিলিয়েন্স, তার কমপ্লায়েন্স, ঠিক আছে, তার মধ্যে কতটা মিনারেলাইজেশন থাকবে, এই যে ফ্যাক্টরগুলো থাকে, ঠিক আছে, অল দিজ আর ফাইনালি অ্যাচিভ, ঠিক আছে, when the functioning is started. বোঝা গেল কিনা? পুরো ব্যাপারটা বোঝা গেল কিনা? গ্রোথ, ডিফারেন্সিয়েশন। গ্রোথের মধ্যে আমরা তিনটে টাইপ দেখলাম, ঠিক আছে, আর এই যে সেলসগুলো রইল তার আমার পোটেনশিয়ালস রইল তিন রকম ভাবে, পার্মানেন্ট সেলস, ল্যাবাইল সেলস অ্যান্ড দ্য কুইসেন্ট সেলস, ঠিক আছে, আর যেগুলো অ্যাক্রিশনারি অথবা অক্সেটিক গ্রোথ রয়েছে, ঠিক আছে, যেমন আবার উসাইট বললাম, নিউরনসে বললাম, ঠিক আছে, দোজ আর সারাউন্ডেড বাই দ্য follicular cells, and in case of neurons these are large axons are supported by the glial cells. All right. Accretionary growth in case of bones and cartilages, where the large amount of minerals are present in the intercellular space. All right. Then comes the differentiation, whereby the tissue potentials, the differentiating potential, the dividing and the differentiation potentials are gradually evolving out. From the totipotent up to the 8 cell stage, we can see the pluripotency, and thereafter we see the histodifferentiation, chemodifferentiation and the histodifferentiation. And final modification, finally we see the molding of organs. All right. And finally it is completed by the when it takes part in the functional activities. বোঝা গেল বডি টিস্যুর পুরো ব্যাপারটা, ফাংশনাল অ্যাক্টিভিটি যখন হবে তখন প্রত্যেকটা অর্গান তার ফাইনাল শেপ আস্তে আস্তে নিতে শুরু করবে। In this process we see the term histogenesis, isn't it? We can observe, we can identify basic or primary 4 types of tissues in our body. That is the connective tissue, muscle tissue, neural tissue and the epithelial tissue. Epithelial tissue, connective tissue, muscle tissue and the nerve tissue. ঠিক আছে? Together with it, this classification I find incomplete and you should write embryonic tissue by the side. 4 basic type of tissues with a special category by its side is the embryonic tissues. If you do not mention embryonic tissues, the embryonic mesoderm is not properly a connective tissue. Embryonic mesoderm, though from the embryonic mesoderm the connective tissues has been derived, but embryonic mesoderm is not a coinage which fits as what we mean by the term connective tissue. কানেকটিভ টিস্যু বলতে যা বোঝায় আর এমব্রায়োনিক মেসোডার্ম বলতে যা বোঝায় দুটো কিন্তু ইকুইভ্যালেন্ট নয়, যদিও কানেকটিভ টিস্যু কোথা থেকে এসেছে, এমব্রায়োনিক মেসোডার্ম থেকে এসেছে। বোঝা গেল? বলো বলো। জাস্ট ওয়েট, কানে আমি খুব কম শুনি, বলো। Very good question. Okay. I understood. No, it is not possible. More at the terminal ages of the cells, cells progeny. Suppose I understood your question and then I appreciate your question. All right. You have asked me whether these cells possess same genetic information as this one. Isn't it? This was your question. All right. Again, you just remember, suppose this is a zygote. This is the zygote. All right. It possesses the phylogeny, genetic information of the phylogeny, the entire phylogeny from the amphibians, the reptiles, the birds and then the mammalian development. All these genetic characters, suppose, are encoded within the genes made up of the structural, made up of the DNA. All these are here. But when they differentiate in a particular lineup, they lose their potentials, which I have already mentioned. ঠিক আছে? This cell could have formed even a zygote of a primate. এখান থেকে একটা প্রাইমেটের জাইগোটও তৈরি হতে পারত। But at the stage of an epithelial cell, suppose this is an epithelial cell. Epithelial cell. ঠিক আছে? This has already translated the genetic information available to this cell line. যে জেনেটিক ইনফরমেশনটা ছিল ইভলিউশনারি, যেটা ছিল ফাইলোজেনি, ফাইলোজেনিটা কোথায়? ফাইলোজেনিটা এইখানে। ঠিক আছে? দিস ইজ দ্য আনরাভেলিং or uncoiling of the information which is expressed here. So this is the ontogeny. This is the ontogeny where there is a blueprint of this generation, not for other generations. Got my point? So all the potentials is not available to a mature form of cells or differentiated kind of cells. ওটা তোমার ছিল একটা সময়, কিন্তু when you are differentiated in a different place, ঠিক আছে, you don't possess the dividing potential or the characteristics which were once in the zygote or at the level of the base cells, not stem cells. Stem cells বললাম না, কিন্তু stem cells বললাম না কেন? Because stem cells are multipotent, not even pluripotent. ঠিক আছে, বোঝা গেল কথাটা? ঠিক আছে, that's why we call ontogeny repeats phylogeny. This is uncoiled. This is uncoiled. Got my point? All right. So at this level, this table of histogenesis, we see four basic types of cellular organizations, that is the epithelial tissue, muscle tissue, epithelial tissue, connective tissue, the muscle tissue and the nervous tissue. To this classification, we will add embryonic tissue. All right. Epithelial tissues will be dealt with elsewhere with the formation and classification of the glandular structures. We come to the topic of connective tissue. All right. Now, in the connective tissue, what do we mean by Peter Muller? As I can remember, connective tissue term was given by a German scientist, Peter Muller. Anyway, you will find it from your books. All right. The basic idea behind connective tissue is that it binds other tissue elements. It binds together the other tissue elements. All right. You will find, if you lift a part of the skin, you will find the dermis and the subcutaneous tissue. You will find the dermis and the subcutaneous tissue. In the subcutaneous tissue, you will find some layers are parallel in relation to the other lamina. মানে কি, একটা সাবকিউটেনিয়াস টিস্যু এরকম রইল, তার মধ্যে ওপরের যে ল্যামিনাটা রয়েছে তার ফাইবারস গুলো যে ডিরেকশনে রান করছে, আদার লেয়ার্স এরও ফাইবারস গুলো সেই ডিরেকশনেই রান করছে। ঠিক আছে, এরকম জিনিস দেখতে পারি। আরেকটা জিনিস দেখতে পারি, we can see another thing, that is the fibers are running in different lamina in different directions. This is the basic idea which I get in identifying tissue sections of connective tissue. এটা আমরা ফার্স্ট থিং দেখতে পাই। ঠিক আছে, ordinarily if you stain tissue section with hematoxylin and eosin, in between the fibers, the spaces in between the fibers are seen as empty spaces. কি বললাম, হিমাটক্সিলিন ইওসিন দিয়ে আমি একটা কানেকটিভ টিস্যু পার্টকে স্টেইন করলাম, we will find only empty spaces in between the bundles of fibers. But freeze-dried preparation stained with special stain, suppose Mason's trichrome, PAS means par iodic acid skip stain, or metachromatically by toluidine blue or Alcian blue, we will be able to find the ground substance as the pink tissues or black by the silver nitrates. So together with the connective tissue, you should read the staining properties of different components of the connective tissues. Connective tissue যখন তোমরা পড়বে তখন তার ডিফারেন্ট কম্পোনেন্ট গুলোকে কি করে স্টেইন করা যায় এবং তার কালারটা কি দাঁড়ায় সেটা কিন্তু মনে রাখবে। ঠিক আছে, আদারওয়াইজ রিডিং কানেকটিভ টিস্যু ইজ জাস্ট ইউজলেস। এটা ছাড়া কানেকটিভ টিস্যু পড়া জাস্ট ইউজলেস। ঠিক আছে, সো স্টেইনিং প্রপার্টিজ তোমরা কিন্তু মনে রাখবে। Suppose some people are very much specialized, but if they run individually, they remain useless. ঠিক আছে? They remain useless also. They cannot attract people or materials which are needed for a big project, তাই না? তুমি ভীষণ ভালো নিউক্লিয়ার ফিজিক্স বোঝো, হ্যাঁ, কিন্তু তুমি রাস্তা দিয়ে, হ্যাঁ, তুমি পকেটে হাত দিয়ে আমি নিউক্লিয়ার ফিজিক্সের সায়েন্টিস্ট বলে, হ্যাঁ, ঘুরে বেড়াচ্ছ, আড্ডা মারছো, হ্যাঁ, এরকম করে ঘুরে বেড়াচ্ছ, তুমি কি কোনো বিগ প্রজেক্টের মেম্বার হতে পারবে? একটা বিগ প্রজেক্ট কি কখনো রান করবে? করবে না, ঠিক আছে? সিমিলারলি ও একজন খুব ভালো কম্পিউটার ইঞ্জিনিয়ার, ঠিক আছে, ও একজন খুব ভালো সেল বায়োলজিস্ট, ও একজন খুব ভালো, ঠিক আছে, হ্যাঁ, প্যাথোলজিক্যাল স্টেনিং টেকনিকও খুব ভালো শিখে এসছে। এইরকম বিভিন্ন যে ইউনিট গুলো, ঠিক আছে, দে আর বেসিক্যালি ওয়ার্থলেস, তাই না? তুমি যখন অ্যাক্টিভিটির রিলেশনে দেখবে তখন দেখবে যে দে আর বেসিক্যালি ওয়ার্থলেস, ওই কাগজের সার্টিফিকেট বগলে নিয়ে ঘুরে বেড়াচ্ছ, তাই না? Until and unless you can be a part of a big system, and that big system is a system, ঠিক আছে, created by the connective tissue. So connective tissue doesn't merely fills up the interstices or the gap in between different tissue elements. বইতে কি লেখা আছে? Connective tissue fills the interstices between the other specialized elements, ঠিক আছে? ছাইবর্ষ বোঝে আর ছাইবর্ষ লেখে। বক্তব্য হলো যে আর্টারি আছে, ভেইন আছে, হেপাটোসাইটস আছে, তার মাঝখানে একটু গুঁজে দেওয়া মেটেরিয়াল আছে, সেটাকে কানেকটিভ টিস্যু বলে। বুঝতে পেরেছো? তো দিস এ ভেরি ব্যাড আইডিয়া। অ্যাকচুয়ালি কানেকটিভ টিস্যু এলিমেন্টস বাইন্ডস অ্যান্ড ইন্টিগ্রেটস আদার এলিমেন্টস, ঠিক আছে, টু মেক হায়ার ফাংশন, টু পারফর্ম হায়ার ফাংশনস অর হায়ার প্রজেক্টস, ঠিক আছে? সো দিস ইজ কানেকটিভ টিস্যু। Ordinarily, we see general or ordinary connective tissue in which we underline number 1 loose areolar tissue, number 2 white fibrous tissue, of which there are dense regular and dense irregular tissues. White fibrous tissue, of which we see dense regular and dense irregular tissues. Then the elastic and the reticular tissues. Together with mentions the adipose tissue, mucoid and the hemopoietic tissues, that is the myeloid tissues. All right. Before going to describing in different forms of connective tissue, you should understand the basic components of a connective tissue. In connective tissue, we have the cellular component and the intercellular components. In intercellular components, we have the amorphous ground substance and the fibrillary structure. So in the intercellular substance, we have two things. Number 1, an amorphous ground substance or the matrix material, and the fibrillar or fibrous component. This is the intercellular component. And in the cellular component, we have two kinds of cells. Cells which are innate to the connective tissue, and cells which are migrant to the connective tissues. ঠিক আছে? কানেকটিভ টিস্যু কিছু মেম্বার আছে যারা ইনেট রয়েছে, আর কিছু মেম্বার রয়েছে যারা বাইরের থেকে এসেছে, লাইক ইন্ডিয়া আর কি, সবকটা বাউন্ডারি ওপেন, যে যেখান থেকে খুশি ঢুকে পড়লো, হ্যাঁ, ভোটার কার্ড, প্যান কার্ড, আধার কার্ড যা চাই হয়ে গেল, ঠিক আছে? ইউ উইল বি আনএবল টু ডিফারেন্সিয়েট, ঠিক আছে, কে আমার দেশের লোক আর কে আমার দেশের লোক। ডোন্ট টেক ইট আদারওয়াইজ, অন্যভাবে নিও না, ঠিক আছে? ডোন্ট টেক ইট আদারওয়াইজ। অল রাইট। সো দিজ আর migrant cells and another is the innate cells. Among the innate cells, we have the fibroblasts, the reticular cells, the fat cells, the pigment cells. All these are innate to the connective tissue. And there are migrant cells like the monocyte cells of the monocyte macrophage system, the lymphocytes, the mast cells, and sometimes the eosinophil, basophil also infiltrate the connective tissue. So these cells are migrant cells, and what I have mentioned, that is the fibroblasts, adipose cells, pigment cells, undifferentiated mesenchyme or the reticular cells, these are the innate cells. So now we have an idea, connective tissue, it has cellular component and the intercellular component. The cellular component has innate cells and the migrant cells. The intercellular component has amorphous ground matrix and the fibrous component. Now I am coming to the ground substance or the matrix material. Alright, in matrix material we see there is a basic structure which is known as the proteoglycans, and there are the structural glycoproteins. There are two components in the matrix part. Number one is the proteoglycans and the structural glycoproteins. Proteoglycans means the repeated unbranched disaccharides. This must be mentioned, alright. Repeated unbranched disaccharides. This change, this which is known as glycosaminoglycans. These are known as glycosaminoglycans. Alright. This is attached or conjugated to a protein. So these are called proteoglycans. Ki bollam? This is conjugated to. Conjugated to a protein. বেশি ঝামেলার দরকার নেই, ঠিক আছে? This is known as glycosaminoglycans. After conjugation, these are called proteoglycans. In the matrix division, matrix or ground substance. This includes number one proteoglycan and the structural glycoproteins. ठीक है जी, proteoglycans are made up of repeated unbranched disaccharide chains, ठीक है जी, which are conjugated to a protein which is known as the proteoglycans. All right, and there is another component which is known as the structural glycoproteins. by the subtypes, we see there are some forms or types of glycosaminoglycans in the tissues. ঠিক আছে? Write down number 1 hyaluronic acid. I'm giving you the properties and other things. All right. The hyaluronic acid, the chondroitin sulfate, dermatan, keratin and heparan sulfate. 3. Dermatan, heparan and keratin sulfate. লিখেছো? hyaluronic acid, chondroitin sulfate A and B, dermatan sulfate, keratin sulfate, heparan sulfate. You have to remember at least one example where we can characteristically see these components. কোথায় আমরা এই কম্পোনেন্ট গুলো ক্যারেক্টারিস্টিক্যালি দেখতে পাবো? Suppose for the keratin sulfate, we will be able to see in the cornea, in the cartilage and in the nucleus pulposus of intervertebral disc. এতগুলো তোমার মনে থাকবে না, হ্যাঁ? কর্নিয়া ফর keratin sulfate, এইটুকুটা মনে রাখবে, কর্নিয়া ফর keratin sulfate, dermatan sulfate in heart, large big vessels, heparan sulfate in mast cells, liver and lungs, mast cell থেকে হেপারিন আসে, ঠিক আছে? লাইক দিস মনে রাখবে তোমরা এক্সাম্পল গুলো, ঠিক আছে? কি কি বললাম? ডারমাটান বললাম, কেরাটিন সালফেট বললাম, হেপারান সালফেট বললাম, বলেছি তো? ঠিক আছে। আর তোমার হাইলুরোনিক অ্যাসিড তো যে কোনো জায়গায় হাইলুরোনিক অ্যাসিড থাকে। হাইলুরোনিক অ্যাসিড, ঠিক আছে, লিভস ইন সলফেট ইন হাইলুরোনিক অ্যাসিড, ঠিক আছে? There are large space which is occupied by the branching filaments, মানে কি বলে, the area occupied by the hyaluronic acid which contains the sulfate groups, carboxylic groups, SO3 minus, ঠিক আছে? আর একটা পজিটিভ চার্জ হচ্ছে সোডিয়াম আয়ন। এই অ্যামাউন্টটা এত বিগ থাকে হাইলুরোনিক অ্যাসিডে, ঠিক আছে, দ্যাট দে বিকামস নেগেটিভলি চার্জ অ্যান্ড অ্যাট্রাক্টস হিউজ অ্যামাউন্ট অফ ওয়াটার। কোনো জায়গায় হাইলুরোনিক অ্যাসিড বেড়ে গিয়েছে মানে সেই জায়গাটায় ওয়াটারি হয়ে যাবে, সেই জায়গাটা it will attract a large amount of water into it, all right, as well as salt, and will give rise to the area in a solfate. তোমরা পড়েছো না যে প্রোটোপ্লাজমের বা টিস্যু আর্কিটেকচারের দুটো স্টেজ হয়, একটা সলফেজ আর একটা জেল ফেজ, তাই না? যখন হাইলুরোনিক অ্যাসিড কনসেন্ট্রেশন বাড়বে, when there is a increase in the hyaluronic acid proportion, the tissue element will go into a solfate, and when it is the chondroitin sulfate or other proteoglycans present, it will enter into a phase of gel phase or dried or dense character. It will attain a dense character. যদি chondroitin sulfate থাকবে, dermatan sulfate, keratin sulfate, heparan sulfate যাই থাকলো, ঠিক আছে, it will give rise to a dense characteristics to the tissue elements. বোঝা গেল? ঠিক আছে। এবারে এই হাইলুরোনিক অ্যাসিডের যে কম্পোনেন্ট, ঠিক আছে, সেই কম্পোনেন্ট কিন্তু আলটিমেটলি আমাদের শরীরের যে ফ্লুইড মুভমেন্টটা সেটা ডিটারমাইন করে। ইট ডিটারমাইন্স দ্য fluid movement in our body. How? তোমরা জানো ফিজিওলজিতে পড়েছো যে তোমার ক্যাপিলারি এন্ড অফ আর্টেরিওল, এইটা আর্টারি, এইটা আর্টেরিওল, এটা ওর ক্যাপিলারি এন্ড, দিস ইজ ইটস ক্যাপিলারি এন্ড, ঠিক আছে? সিমিলারলি দিস ইজ দ্য ভেনাস এন্ড, this is the venular end, this is the sides of anastomotic channel. There are cells in between. ঠিক আছে? আর্টারিওলার এন্ড অফ ক্যাপিলারি প্রেসার কত? কে বললো? 32 বললে? ফাটাফাটি। আজিস কোথায় বাবা? ফার্স্ট বেঞ্চে বস। কোথায় কি পিছিয়ে আছিস, লুকিয়ে আছিস? আয় বাবা সামনে বস, মুখ দেখে পড়াতে ভালো লাগে। আয় সামনে আয়, তুই আর পেছনে বসবি না, আয় সামনে বস, আয় আয় আয়, ঠিক আছে? আয় আয় সামনে বস, সামনে বস। So this is 32, this pressure is 32 millimeter of Hg. All right. The plasma here gives rise to 25 millimeter of Hg. Colloidal osmotic pressure. All right. And at the venular end, this is 19 millimeter of Hg. So at the venous end, fluid can go in, and then the arteriolar end, fluid will go out. এই যে আমরা টিস্যু প্রেসার এবং অ্যামরফাস সাবস্ট্যান্সেস মানে হচ্ছে প্লাজমাতে যে প্রেসারটা আমরা ডিনোট করলাম, ঠিক আছে? These are mostly determined by the connective tissue elements. That is the matrix material that determines the এই যে প্রেসার, ঠিক আছে? টিস্যু প্রেসার as different component, ঠিক আছে? This is primarily determined by the connective tissue elements, ঠিক আছে? যদি হাইলুরোনিক অ্যাসিড বেশি থাকে তাহলে প্রচুর জল জমে থাকবে, ঠিক আছে? যদি অন্য এলিমেন্টস গুলো থাকে অত বেশি জল থাকবে না, ঠিক আছে? The next thing is when we examine the collagen fibers under the microscope, we do not understand that collagen fibers can be made up of finer fibrils. কি বললাম কথাটা? মাইক্রোস্কোপের নিচে যখন কোলাজেন বান্ডলস গুলো দেখছি, ওয়েভি বান্ডলস গুলো যখন আমরা দেখছি, তখন কিন্তু আমরা বুঝতে পারি না যে দিস আর মেড আপ অফ ফাইনার ফিব্রিলস। প্রত্যেকটা সুতোর মধ্যে যে আরো পাতলা পাতলা সুতো আছে সেটা আমরা বুঝতে পারি না। হাউ ক্যান আই আন্ডারস্ট্যান্ড দিস? কি করে বুঝলাম আমরা? ঠিক আছে? when we examine the collagen fibers under polarized light, ঠিক আছে, two separate beams are refracted out in different directions, ঠিক আছে? two separate beams are refracted out at different directions. This phenomenon is known as birefringence. এই শব্দটা লিখে রাখো, শিখেও রাখো জিনিস, ঠিক আছে? This is known as birefringence. So this is the birefringence. Property of birefringence, which gives us the idea that the larger fibers of collagens are made up of finer fibrillar structures. B-I-R-E, birefringence, F-R-I-G-E-N-C-E, birefringence. So staining properties, the metachromatic dye is the Mason's trichrome, Alcian blue, Toluidine blue. All right. Periodic acid skip 10, there is for pass, and there are freeze dried preparations, all I have mentioned. ঠিক আছে? And you will read it more particularly. All right. Without knowing this thing, this reading is useless. Then the components of the types of the glycosaminoglycans glycans available to different tissues. And the next thing comes is the structural glycoproteins, of which the laminin, osteonectin, fibronectin, chondronectin, these are available, যেগুলো তোমরা ফিজিওলজিতে পড়ে নেবে। ঠিক আছে? তাহলে এটা আমার স্ট্রাকচারাল কম্পোনেন্টের দিকে হয়ে গেল, এক্সেপ্ট দ্য ফাইব্রাস কম্পোনেন্ট। অলরাইট, ফাইব্রাস কম্পোনেন্টের মধ্যে বাকিগুলো পরে বলবো, একটুখানি কোলাজেন বলি, শেষ হয়ে এলো, টাইমটাও শেষ হয়ে আসছে। ঠিক আছে? Collagen fibers are 1 to 12 micron in diameter. ঠিক আছে? And these have no side branches. ঠিক আছে? Individual collagen fiber has no side branches. ঠিক আছে? 1 to 12 micron in diameter. These are made of finer fibrils of 20 to 200 nanometers. ওগুলো আবার যে ফিব্রিলারি স্ট্রাকচার দিয়ে তৈরি, সেগুলো 20 to 200 স্ট্রাকচারস। ঠিক আছে? এগুলোকে বলে ট্রোপোকোলাজেন। ট্রোপোকোলাজেন। ঠিক আছে? ট্রোপোকোলাজেন, 3টে করে ট্রোপোকোলাজেন মলিকুল এক জায়গায় হয়ে প্রোটোকোলাজেন, সরি 3টে করে প্রোটোকোলাজেন মলিকুল এক জায়গায় হয়ে ট্রোপোকোলাজেন। ঠিক আছে? কোলাজেন সিনথেসিস বায়োকেমিস্ট্রি আলাদা করে পড়তে হয়। ঠিক আছে? উইদিন দ্য কোলাজেন মলিকুল উই সি হাইড্রক্সিপ্লোরিন, হাইড্রক্সিলাইসিন, ঠিক আছে, গ্লাইসিন অ্যান্ড ভ্যালিন অ্যাজ দ্য মোস্ট ইম্পর্টেন্ট অ্যামিনো অ্যাসিড। গ্লাইসিন, ভ্যালিন, হাইড্রক্সিলাইসিন অ্যান্ড হাইড্রক্সিপ্রোলিন। যখনই কোনো জায়গায় কোলাজেনাস টিস্যু, ধরো তোমার মাসেল ব্রেকডাউন হচ্ছে, ঠিক আছে, ওয়েট লস হচ্ছে, মাসেল ব্রেকডাউন হচ্ছে, ঠিক আছে, ইফ ইউ মেজার ইউর ইউরিন there will be increased amount of hydroxyproline residues, underline করো, ঠিক আছে, whenever there is breaking away of collagenous fibers in different tissue components, we will get high level of hydroxyproline in our urine. And hydroxylysine sites are the sites of cross linkages between different fibers. Hydroxylysine residue গুলো যেখানে আছে সেখানে ক্রস লিঙ্কড হয়, ঠিক আছে। এবার বডির একটা খুব পিকুলিয়ার প্রপার্টি দেখবে তোমরা যখন বোনস পড়বে, ঠিক আছে, বোনসের মাসল দেখবে, আগের কম্পোনেন্ট, ধরো স্ক্যাপুলা পড়েছো, ঠিক আছে, স্ক্যাপুলা পড়েছো, স্ক্যাপুলা থেকে যে মাসলগুলো স্টার্ট করলো, ঠিক আছে, স্ক্যাপুলা থেকে যে মাসলগুলো স্টার্ট করলো, ধরো ওপরে এইখানে থাকে সুপ্রাস্ক্যাপুলারিস, ঠিক আছে, এইখানে তোমার সুপ্রাস্পাইনাটাস, এখানটা ইনফ্রাস্পাইনাটাস, স্ক্যাপুলার এই যে পেছনটায় এই কম্পোনেন্টটাকে বলে সুপ্রাস্পাইনাটাস কম্পোনেন্ট, এইটা ইনফ্রাস্পাইনাটাস কম্পোনেন্ট, মাঝখানে একটা স্ক্যাপুলার ক্রেস্ট আছে, ঠিক আছে, একটা ডিভাইডিং একটা বোন আছে, ঠিক আছে, হরিজন্টাল প্লেট অফ বোন আছে, ঠিক আছে। এই যে সুপ্রাস্পাইনাটাস এর ইনফ্রাস্পাইনাটাস, যেটা প্রাইমারিলি অরিজিনেট করলো কোত্থেকে? স্ক্যাপুলা থেকে, ঠিক আছে। দিস হ্যাজ টু বি ইনসার্টেড within the proximal third of the humerus, the next bone. ও কোথার মধ্যে ইনসার্ট করে যাবে? প্রক্সিমাল থার্ড বলি, অ্যাকচুয়ালি প্রক্সিমাল ফোর্থ, ঠিক আছে, তার মধ্যে ইনসার্ট করে দেবে, ঠিক আছে। নেক্সট লাইন অফ মাসলস will originate thereafter. মানে সোজা কথা হচ্ছে একটা গ্রুপ অফ ফাইবার্স, ঠিক আছে, তারপরের গ্রুপ অফ ফাইবার্স, তারপরের গ্রুপ অফ ফাইবার্স এরকম করে স্ট্যাগার্ড ফ্যাশনে অ্যারেঞ্জ থাকে কোলাজেন ফাইবার, ঠিক আছে। মানে আমরা যদি দেখি যে একটা মাসল এইখান থেকে এইখানে আছে, তাহলে কি ওর ফাইবার্স, কোলাজেন ফাইবার্স গুলো এখান থেকে এরকম করে আছে টোটাল, ঠিক আছে, না, এই ফাইবার্স গুলো এরকম করে স্ট্যাগার্ড ফ্যাশনে রয়েছে, ঠিক আছে। পিরিয়ডিসিটি থাকে একটা, হ্যাঁ, 2800 আর্মস্ট্রং এর মধ্যে 670 ডিগ্রি আর্মস্ট্রং এর একটা পিরিয়ডিসিটি থাকে, অর ক্রস ট্রাইসেন্স থাকে, ঠিক আছে। অ্যান্ড দ্য ওভারল্যাপিং ইজ রাফলি 1/3 to 1/4। সেজন্য হোল বডিতে তুমি যেখানেই দেখবে এই যে হিপ মাসলস গুলো, ঠিক আছে, এসে কি হবে, গ্রেটার ট্রকেন্টার, লেজার ট্রকেন্টার, আপার থার্ড অফ ফিমারের মধ্যে অ্যাটাচ হয়ে যাবে। এখান থেকে স্ক্যাপুলার মাসলস এসে, ঠিক আছে, এই যে হিউমেরাস, পড়েছো, বোন পড়তে শুরু করেছো? হ্যাভ ইউ স্টার্টেড রিডিং বোনস? না, ঠিক আছে। এবার এইখানে তুমি যদি হিউমেরাসটা, এখানে যদি একটা হিউমেরাস আঁকো, এই যে জায়গাটা আঁকলাম, সুপ্রাস্ক্যাপুলার, ইনফ্রাস্ক্যাপুলার আর টিরিস মাইনর, ঠিক আছে। এইখান থেকে মাসেল গুলো সুপ্রাস্পাইনেটাস মাসেলটা এইখান থেকে এইখানে চলে যাবে, ঠিক আছে, ইনফ্রাস্পাইনেটাসটা এইখানে যাবে, আর টিরিস মাইনর এখানে আছে, সেই টিরিস মাইনরটা যাবে, ঠিক আছে। এটা বললাম কেন? দ্য মাসেল ফাইবার্স or the collagenous tissue that is being originated in the proximal part will end up within the upper 1/3 or 1/4 of the tissue of the next flow of or next band of collagenous fibers. এটার অন্যথা কোথাও দেখতে পাবে না, এটার অন্যথা তুমি এক্সেপশনস তুমি দেখতে পাবে না, বুঝে গেল? দিস ইজ দ্য স্ট্যাগার্ড ফ্যাশন অফ অ্যারেঞ্জমেন্ট অফ কোলাজেনাস ফাইবার্স, ঠিক আছে। অনেক কথা বলবো আমি কোলাজেন ফাইবার্স নিয়ে, ঠিক আছে। এখন বাই মডার্ন টেকনিক্স উই হ্যাভ আইডেন্টিফাইড অ্যারাউন্ড 28 টাইপস অফ কোলাজেন, অফ হুইচ ইউ শুড রিমেম্বার হার্ডলি 5 or 7। তোমরা 5 থেকে 7টা টাইপ মনে রাখলেই হয়ে যাবে, ঠিক আছে। সো কোলাজেন ফাইবার্সের টাইপস একটু মনে রাখো, ঠিক আছে। ফার্স্ট লেখো এরকম করে যে ফিব্রিলার অ্যান্ড নন ফিব্রিলার, ঠিক আছে। রাইট ডাউন ফিব্রিলার অ্যান্ড নন ফিব্রিলার কোলাজেন ফাইবার্স। ফিব্রিলারের মধ্যে থাকবে 1, 2, 3, 5, 11। 1, 2, 3, 5 লিখলেই হয়ে যাবে তোমাদের, 11টা জাস্ট বলার জন্য বললাম, ঠিক আছে। আর নন ফিব্রিলারের মধ্যে থাকবে টাইপ 4 বেসমেন্ট মেমব্রেন কোলাজেন টাইপ 4, মাইক্রোফিলামেন্টাস মানে হচ্ছে তোমার টাইপ 6, অ্যাঙ্করিং ফিব্রিলস টাইপ 7, ঠিক আছে। মাইক্রোফিব্রিলস, অ্যাঙ্করিং ফিব্রিলস টাইপ 6 অ্যান্ড টাইপ 7, আর বেসমেন্ট মেমব্রেন হচ্ছে তোমার টাইপ 4, ঠিক আছে। আর শর্ট চেন চাপে ফেললে, শর্ট চেন, আচ্ছা 8 লিখে রাখো, বাড়িতে গিয়ে দেখবো, ঠিক আছে। শর্ট চেন লিখে দেখো 8, টাইপ 8 অ্যান্ড টাইপ 10 লিখে রাখো, ঠিক আছে। আই উইল ক্রস চেক, ঠিক আছে। হ্যাঁ বলো। ফিব্রিলার অ্যান্ড নন ফিব্রিলার, ঠিক আছে। ফিব্রিলারের মধ্যে রয়েছে টাইপ 1, টাইপ 2, টাইপ 3, টাইপ 5 অ্যান্ড টাইপ 11। নন ফিব্রিলারের মধ্যে বললাম, ঠিক আছে, মাইক্রোফিলামেন্টাস যেটা বললাম তোমার টাইপ টাইপ 6, অ্যাঙ্করিং ফিব্রিলস বললাম টাইপ 7, তাই তো? বেসমেন্ট মেমব্রেন বললাম টাইপ 4, শর্ট চেনে বললাম 8 আর 10, তাই না? আর দুটো ভ্যারাইটি এমনি মনে রাখতে পারো, নাও মনে রাখতে পারো, দ্যাট ইজ দ্য কি বলে ফিব্রিল অ্যাসোসিয়েটেড ফ্যাসিড আর ম্যাসিড, মানে মেমব্রেন অ্যাসোসিয়েটেড, ঠিক আছে, কোলাজেন ফাইবারস, আর হচ্ছে তোমার ফিব্রিল অ্যাসোসিয়েটেড কোলাজেন ফাইবারস, ম্যাসিড আর ফ্যাসিড, ঠিক আছে? মনে রাখতে পারো, নাও রাখতে পারো, ঠিক আছে, ইট ডাজন্ট ম্যাটার, ঠিক আছে? MACIT ম্যাসিড অ্যান্ড ফ্যাসিড, FACIT, এই দুটো টার্ম মনে রাখলেও হবে, না রাখলেও হবে, ঠিক আছে? অত কিছু দরকার নেই, ঠিক আছে? আউট অফ দিস, ঠিক আছে, প্রিডমিনেন্টলি দেয়ার আর টাইপ 1 কোলাজেন, predominantly there are type 1 collagen in bones and fibrocartilages. This is almost 90% and constitutes 25% of our body protein. ঠিক আছে? মানে 25% of our body protein is collagen protein, of which 90% is type 1 collagen. ঠিক আছে? Constituting our bones and cartilages, especially the fibrocartilages. All right. Type 2 is the hyaline cartilage. তাহলে টাইপ 1 হয়ে গেল, টাইপ 2 ইজ দ্য হায়ালিন কার্টিলেজ। হায়ালিন কার্টিলেজ কোথায় থাকে? যে কোনো গ্রোয়িং এন্ড অফ লং বোনস দেয়ার ইজ হায়ালিন কার্টিলেজ। আর যে আর্টিকুলার কার্টিলেজ, মানে নি জয়েন্টের এখানে টিবিয়া, এখানে হিউমেরাসের কন্ডাইল, এই দুটো কন্ডাইলের মাঝখানে যে ক্যাপ থাকবে, ঠিক আছে, আর্টিকুলার ক্যাপ ইন জয়েন্টস আর মেড আপ অফ হায়ালিন কার্টিলেজ। ইন গ্রোয়িং পার্ট অফ লং বোনস হায়ালিন কার্টিলেজ, and in the articular caps there are hyaline cartilage which are made up of type 2 collagens. All right. Type 3 collagens are in reticular tissues. ठीक है जी? They forms a network to hold the cells of glandular organs. This type 3. Type 4 is the basement membrane collagen. Type 4 is the basement membrane collagen. And type 5 is the placental tissue. ঠিক আছে? তোমাদের জন্য এই 5টা মনে রাখলে হবে, 6টা বা 7টা যদি মনে থাকে ইটস অল রাইট, না হলে এই 5টা টাইপ মনে রাখলেই হয়ে যাবে। ঠিক আছে? নাওএডেজ পরে আসছি, অন্য জিনিসগুলোতে পরে আসছি। কটা বাজে গো? হ্যাঁ, চলো। কোলাজেন নিয়ে আরো পরে অনেক গল্প হবে, অনেক কথা বলার আছে, ঠিক আছে? কারণ এখন তো কোলাজেন পাওয়া যায়, জানো তো? কোলাজেন মানে এমনি কোলাজেন পোরসাইন, ইকুয়াইন আর বোভাইন রিসোর্স থেকে, পোরসাইন, ইকুয়াইন আর বোভাইন রিসোর্স থেকে কোলাজেন ফাইবারস আনা হয়, ঠিক আছে? বোন গ্রাফট হিসেবে ইউজ করে। 1 মিনিট, কিপ সাইলেন্স, একটু আসতে, একটু আসতে। বোন গ্রাফট হিসেবে, ঠিক আছে, উন্ড আর বার্নে, ঠিক আছে, উন্ড আর বার্ন ড্রেসিং অ্যাজ ওয়েল অ্যাজ রিকনস্ট্রাকটিভ সার্জারিতে কোলাজেন ফাইবারস ইউজ, ঠিক আছে? আমাদের নাকটা পচা আছে, আমার নাকটা একটুখানি তুলে দিন, এই যে প্লাস্টিক সার্জারি, ঠিক আছে, তাও সেটাও ডিপেন্ড করে হচ্ছে তোমার এই কোলাজেন ফাইবারস এর উপর, ঠিক আছে?
That means all of you know Bengali.
Yes.
Okay. So last class, what we have discussed? Take a class, don't tell anybody. Anyone? Last class, what was their class?
About blood and the way we can we can collect blood from fingertips and veins actually, and the different tests.
Okay, collection of blood samples.
Yeah.
Okay. So today we'll start that whatever is collected, how we'll preserve and how we'll process and transport also. So preservation of the sample. Okay. And processing also. Storage and processing. Storage and processing. processing of blood sample. So I told how we collect. How we collect blood sample by which test tube? Which test tube? That means anticoagulants is required. Suppose you will collect blood sample for blood sugar estimation. Blood sugar estimation. That test tube contains what kind of anticoagulants? Sodium chloride and potassium oxalate mixture is there. Okay. So what is the role of why you will collect only sodium chloride tube? I told last class why we will collect in sodium chloride tube only. Anybody? That will collect in sodium chloride tube only. Sodium chloride tube, why? What is the role?
It's anticoagulant.
Yes, it is anticoagulant. Why not heparin?
Sir, it inhibits enolase, that's inhibit
Yes, that sodium chloride, it inhibits the enolase enzyme. So, what will happen? Whatever you have collected in sample, that will be actual calculation, or that means glycolysis will be prevented. The process of glycolysis will be prevented by the sodium chloride. Okay. Suppose you want collection for ABG, arterial blood gas analysis, ABG analysis. For that, what kind of anticoagulants required? For that, heparinized tube. You will through heparinized tube. We will collect it for ABG, arterial blood gas analysis. Okay. That tube should contain heparin. Arterial blood gas. Arterial blood gas. ABG. Okay. And one thing you have to keep in mind. Suppose you are collecting bilirubin. You are collecting sample collecting for bilirubin test. That what precautions you have to take? What precaution? That is very important. Bilirubin will be destroyed by light. Light sensitive. Bilirubin is light sensitive. So what will happen if you collect blood sample and you will more light is there, or in sunlight when you are transporting the sample through sunlight you are going for sending the sample. What will happen? That ultraviolet light will destroy the bilirubin. So the concentration of bilirubin will be decreased. So that you have to keep in mind. That means bilirubin is light sensitive, so avoid light. Next is here, suppose you are collecting for electrolytes. What are electrolytes? Name the electrolytes? Sodium, potassium chloride. Suppose you are collecting for that. That sample should not be hemolyzed. That is very important point. First thing is, any sample you are collecting, that should be fresh sample. That should be always fresh sample. If suppose you require, later you will do for test. Later you will do for test. 2 days or 3 days you want to keep that sample. That you can at 4 degrees centigrade you can keep that sample for further processing, for further requirement. Okay. So if that sample is hemolyzed, that avoid that sample for all tests. First thing, very important point. Hemolyzed sample always avoid. And out of that, if you are going for electrolytes test, sodium potassium chloride, that hemolyzed sample should not be used. Okay, electrolytes should not be used for the hemolyzed sample. Next, these are all tests. If you will not do test, you will require further, then you can keep in fridge for at 4 degrees centigrade. Next thing is, what are the Safety purpose. What is biological safety? Biological safety. Okay. For that biological safety, what you should do? When we enter to the lab, practical lab, two things very important. One is with how we enter. Your dress should be proper. Your dress should be proper. That means what is that? Lab white coat. White coat should be there. And second thing, yes, goggles you can utilize when at what time if any reaction occur or reactive materials is there. Okay. Next. Covered, yes. Full shoe. That is very important because you are using their test tubes, glass bottles, glass wires are there, reagent bottle. Okay, reagent bottle, mouth pipette is there. All these things. Suppose you should take care. Suppose you are keeping that reagent bottle, you are taking some reagent, that time it will fall, breaks. So it may injury. So always don't come in the half chapel. Okay, always. So here it should be full, so that is good. It can prevent. If a reagent may fall, chemicals or hazardous material, it may fall. So it can protect you. Okay. Next.
Gloves.
Huh? Gloves. Very important thing. Gloves. You should use. Gloves should be. Why? Because that is also it will protect your hand. Any reagents or any chemicals, it can protect. Next, what we do? Yes, that is very important thing. Suppose in Bunsen burner you are doing any test and long hair is there like that. It can burn. So always hair should be tied. Yes, that is one point, important point. Hair should always tie. Next. Yes, tell. Nails. Another any important thing, chocolates or any food materials that don't take in the practical area. Okay, food materials it will be prevented, protected. Don't take any food materials. And sometimes what will do? The time is not there. Immediately with the teacup you will run from hostel to practical lab. That habit should not be. Any other?
Sir, why are all the gloves and coat everything white? Why not some other color?
And that I guess that I have to see. Why are we this color? Okay, next. Other things. Okay. Hello. Next, any other things? Very important thing is that whenever instruction is not given to start practical, that you should obey. Instruction before given instruction, you should not start practical. Instruction will start practical. Whatever instruction given, like regarding any practicals, any test, you don't start. Second thing, any other things?
Practical rules, sir.
Very important. Practical record with your demonstration copy. How many of all of you brought or not?
Demonstration.
One is demonstration copy, another is a practical copy. Next, any other?
The testing board.
Testimonial is there. Already there. That should not, yes. Always use only testiholder. Don't with hand, direct don't pour. Next. Mass also you can use. Any other?
People died in Kolkata due to COVID.
And very important thing is there, if any poisonous chemicals is there, or reagents is there, suppose some reagents are there, they are carcinogenic. Suppose benzidine, we are doing blood present or absent in urine. That for that we can do benzidine test. Benzidine. Benzidine test. That time, what to do? Mouth bed will be prevented. Don't go for mouth bed. Okay, benzidine is why? Because it can cause cancer. Okay, benzidine is carcinogenic. Next, any other? So any chemical, any reagent, don't go for mouth bed. Okay, don't go for mouth bed. What are acids? What acids we can utilize in laboratory? What are strong acids? H2SO4, HCl, nitric acid, acetic acid, uric acid. So all these acids, when you utilizing, that should not go for mouth bed. Suppose in mouth bed also, in urea test for urea, one acid mixture is there. Okay, suppose that time we are mouth bedding. Okay, then what to do?
Proper use, proper.
Stopper you can use. After that also, somebody are there, they will open them. It is not coming properly. Mouth bed, they will do mouth bed. Then immediately you should, first thing is wash your mouth by normal water. Next.
Any basic?
Yes. Basic. That means alkali solution. Maybe sodium carbonate. Okay, 5% sodium carbonate. That solution you should immediately, with that you should gargle. Okay. Next, that sodium carbonate solution also you can utilize. You can use here, lip and mouth also. If suppose alkali, what are alkalis are there? NaOH barium hydroxide, then ammonium hydroxide. Suppose all these alkalis when you are using, that time suppose by mouth bedding it will go. Then what will do? Acid.
Weak acid.
Weak acid. That weak acid. Okay. Best is your lemon juice or vinegar.
Citric
Citric acid also.
Vinegar.
That will be utilized. That also same thing by gargling, and also you can utilize your lip and mouth. Then other things.
So can't we avoid using mouth fillers?
Huh?
Can't we avoid using mouth fillers?
Is
Ah,
there
yes.
an alternative for mouth fillers?
Yes, stopper is there. Rubber valve is there. That also you can utilize. Mouth bed not required. Because what happened? Particular when you go for mouth with rubber valve, you accurate. That practice required for accuracy. But mouth bed easily it will suction. How much it is coming? You can see high level up to 5 level. Now stop. Okay, well. Now one thing is that suppose all hazardous material, how will dispose? How? Once you practical area, after finish your practical, Okay, how it will be that hazardous material? How, suppose acid you are using that acid. Now, if test tube acid is there, that you will throw
We have to wash the
In
washing
running-
machine.
wash basin.
In running water.
Running water. That means first, what is that? Yes, that is with running water you will use. Why? Because first that acid should be neutralized. That means what you will use? Dilute that acid, then you throw it. Same for acid, same for also alkali. Okay. Next is here anticoagulants. What are anticoagulants we are using? One is heparin. Another is the EDTA. EDTA also. Ethylene. EDTA will be used like any other. Sodium citrate. What are some oxalates? These are chlorides. All these things that here anticoagulants will be utilized. Okay. Then any other things? Regarding chemicals, in chemistry practical what you did? Once it is here, where you are doing practical, that is bench step. One is side cell. Side cell reagent is there and also bench step reagent is there. That means where you are using or you are doing practical, that is bench step. What reagents will you keep there? Only those are required. That is the correct only. Those are required. Non-required things why you keep there? Those are required only you will keep. Okay. So mostly what you do? Those are strong acid. Okay, strong acids will not keep in your bench top area. That means practical doing area where it will do that area it should not be there. And what other things? Other things means one test tube, a rake will be there. Test tube rake or test tube stand. Then test tube. Another thing, holder, test tube holder will be there in bench top. Another, one set burner will be there. Very good. Any other? Pipette. One is glass pipette. Another is your auto pipette. Both will be there. Any other? Beaker will be there.
Distilled water.
Distilled water, yes, distilled water will be there. And another thing, after finish all, that bottle dispenser should be there. Bottle dispenser. That means water is there. That wash your test tube, you can leave practical. Okay. What reagents will keep there in side cell?
Concentrated acids.
Concentrated acids and
Light sensitive materials. Light sensitive reactants.
yes, light sensitive material is not like that.
AgNOx silvanite.
Acetonate, yes. And another thing important is there. Suppose that any reagents it cause fumes. Any reagents it can cause fumes. That reagents you have to protect. How that where you will do that reagents utilize that will in fume compound. That will be utilized in fume compound. Okay. Next. One is a bench step, another is a side cell. And what instruments will be there? In side cell instruments?
Droppers.
Side cell instruments. One is your water bath,
Flask. Conical flask.
conical flask, water bath, hot water bath. Anyone? Any other? Colorimeter? Not calorie. Colorimeter. pH meter very important. pH meter will be in the side cell. Any other? Spectrometer is also side rack.
Incubator. Incubator.
Incubator, yes. Any other? Periods. Any other? Centrifuge, very good. Side cell. Any other? Weighing machine, yes. Any other? Another important thing we are forgetting.
Weight machine.
Contact cell, yes. Any other? Thermometer, yes.
Weight machine. Microwave.
Any one that is very important. But whatever you are listen, whatever you are utilizing or you are using the test tubes, first you should dry. For that only important is that is hot air oven. You after use wash it and it will put in hot air oven. That should be dry. Okay. Any other? So next class onwards we'll start your normal constituents of urine. Okay, normal constituents of urine. Next class we'll start. Okay. So this is your next class. Normal constituents of urine. What are the physical appearance? What color should be there? Okay. What is the special gravity?
Next class will be in January because from Monday there is an exam.
Yes.
And next class will be next month.
Sir, after
Then notice. Okay. Any other things?
Sir, can we visit
We visit there. Okay, we can visit. But science says, it a key area. What is the area? That area? That is students area. Your area. What I told, whatever is required for the practical, that means always you will get your test tube, rack. Okay, that is boiling water. We go where? We go for. You may do for bending test in Bunsen burner. Also, you can do in hot water bath. In 2 minutes, we will keep boiling water bath. That also you can get positive. Okay, and estimation of urea, quantitative estimation. That time also that hot water bath you can utilize.
This college, okay. Now, yesterday few of you actually met, with whom I had some sort of introduction, okay, but many of you are absolutely new to me and myself is also new to you also. Now, we have come across about the general features of bones and classification of different types of bones and classification of bones. Okay. Now we will going to start the osteology of the lower limb. Osteology means
Sir, आगे जो हमने मैम पढ़ी तो ওটা কি পরে কন্টিনিউ হবে ক্লাসটা আমাদের
joints
as per our HOD's command, we are going to start from today onwards the bones of the lower extremities. Okay. I am being directed by the HOD. So, if there is any confusion, you can have a discussion with HOD. Now, from today onwards, we will start the bones. Rather, today I will try to give us introduction about the bones of the lower limb. Now, there are some bones which are, we know the vertebra, this is the sacrum, okay, and sacrum actually it is we will know in detail in our abdomen class. Okay. Now, this bone is a hip bone which articulates with the sacrum. Okay. To form a joint which is called the sacroiliac joint. Okay. And remember, all these joints are very, we will actually bears the weight. So there are, okay, so all these, or this joint actually, it is actually bears the weight of the body. Okay, so, and it articulates like this. And on either side, we will get another hip bone, and which will articulate in the middle to form a joint, which is a secondary cartilaginous joint. Okay. The secondary cartilaginous joint, why it is second? What are the criteria or features of the cartilaginous joint? We will know later on. But just remember the term. This is a secondary cartilaginous joint. The joint where the movement is restricted, but there is some movement. In fibrous joint, in between the skull bones, the movement is absolutely not possible. Okay. But in cartilaginous joint, secondary cartilaginous joint, there is a big movement. Okay. So this is a symphysis pubis or symphysial joint. Okay. Now these two bones along this sacrum, okay, forms the pelvic girdle. So, two hip bones along with sacrum behind, okay, this becomes one entity which is called the pelvis, or the bones, this which after articulation we call it as a pelvic girdle. Similarly, in our superior extremity, there is another girdle which is called the pectoral girdle. And there are two bones which forms the pectoral girdle. They are the scapula and the collarbone of the girdle. One is behind, which is present over the ribs, which is also called the shoulder blade or the scapula. Okay. But this pelvic girdle is very much stable, and it bears the major weight of the body, because we know this is the femur. Okay. The weight of the body actually goes through through our axial skeleton, and ultimately it is transmitted through this joint or the hip joint. The half of the weight through one hip joint, half of the weight of the body through the another hip joint. So you can appreciate the weight of when the weight of the body is carried down through this joint. So this pelvic girdle must be very much stabilized. Okay. So to stabilize this joint, there this sacroiliac joint, you will know that this is very much stable, and it is being articulated by some ligaments. There are some ligaments. There are many factors. One of them is the ligament. There are anterior or intermediate, and another is posterior sacroiliac ligaments. Okay, which helps to bind these two bones. Okay. But in the pectoral girdle, there is actually the weight is carried through the collar bone, and ultimately it is carried to the axial skeleton. Okay, actually weight of the upper limb is carried through the collar bone. So it is, and there is a continuous movement, and there is the joint cavity in the scapula is very small because of the fact it is a nominal weight bearing. But here the weight, you can see the joint space is more, so that this joint is very much stable. Okay, and this is the difference between the pectoral girdle and pelvic girdle. And the bones are, and the articulation is much more stable. To maintain the stability, you can see the wide joint space, the cup-shaped area. This cup-shaped area is called the acetabulum. Meaning of the acetabulum is cup or peg. So this is acetabulum. Whereas in our shoulder joint, there is in the scapula there is small glenoid cavity, so that all the movements are very much easy to perform. When the mobility is more, stability is less. So this shoulder joint, actually frequently there is a sublocation or dislocation of the shoulder joint, whereas this joint is very stable. So it is sublocation or dislocation is relatively less common. It is not that there will be no dislocation. It happens in many motorcycle accidents and many cases, or in congenital dislocation of hip in small child. We can see. Okay. Now we will come. Now this is the hip bone. Now next bone is the femur. We can see this is the longest bone of the body. Okay. Do we know the long bone and short long bone? This is the long bone, which has got upper end, lower end, and intervening shaft. Okay. Then we can see the tibia. Okay. So it is not matching with this one. This is not of this side. This is rather on the other side. Just forget the lower end. This is the tibia, and along with there is another bone which is stick-like bone. This is the fibula. Okay. Then in the lower extremity we get the articulated skeleton of the foot, where we can see there is an articulation between the tibia and this bone. This is called the talus. This is the talotubular joint, or the part of the ankle joint. Okay, talotubular joint. Okay. Now the keystone on which we base our foot, this is the calcaneum. Just this is an overall introduction. Just try to identify the bones. So this is the talus and this is the calcaneum. Next, in front, we are getting a boat-shaped bone, which is called the navicular. Okay. And there is a joint between the talus and the navicular. Okay. This is the head of the talus. Okay, which joins with the navicular, and this joint is called. Actually, the joint is also related with calcaneum. Calcaneum navicular joint. This is the calcaneum navicular joint, and there is a one very important ligament which is called the spring ligament, which helps to maintain the arch. And this is a and which is called the spring ligament, and which maintains the medial arch. You can see there is an arch, and that is maintained by one ligament. Okay, that is the talocalcaneonavicular ligament or spring ligament. Okay, which maintains the medial arch of the foot. Okay. Now, the next set of bones, we can see they are three bones from medial to lateral, medial, intermediate, and lateral cuneiform bones. And most laterally, we can see the cuboid bone. Okay. So these bones actually they are forming the and in front of these bones we are getting another set of bones which are actually the metatarsals. All these bones are tarsal bones. You can see the 7 tarsal bones, and in front we are getting the metatarsal bones. Phalanx এখনো যাচ্ছি না. Okay, so confine করেছি up to this is the metatarsal. And then we can find the phalanges. We can find the 3 phalanges. We can find 3 phalanges laterally on the lateral 3 or 4 digits, but there are 2 phalanges in the great radius. Okay, so these are the different bones of the inferior extremity. Now I will confine myself to this hip bone. Now, first of all, we must know one thing, that is the anatomical position of a bone. Okay. Before that, I want to impress something, that is, this is within the hip bone, there is one cup-shaped area which is present laterally. Which is called the acetabulum, and all the other parts are oriented to this acetabulum. The one part you can see is the fan-shaped area which goes above. This is the ilium. So it is present above and behind the acetabulum. One part, you can see this one, this is oriented in front and below. This is called the pubis. And another part which is in relation, this is below and behind, this is the ischium. So, grossly there are 3 parts. This is the fan-shaped expanded flat part, which is called ilium. This one is pubis, and this is the ischium. But now we will start actually from after a few days we will start the dissection of the lower extremity. Okay. And there are many muscles which originate in the trunk. They are attached to this bone. And there are some muscles which originate from this bone and ultimately they are inserted over the bones of the lower limb or lower extremity or inferior extremity. Okay. So we are mainly now onwards we will be confined our discussion with the muscles or any ligament which are originating from this bone and going towards the inferior extremity. So this is the surface, we can see, this is the outer surface and this is the inner surface, okay, of this hip bone. The outer surface is actually, this is the outer, or actually this area is the femur, and there are all the muscles, they are actually related to the femur. So we call the outer surface or the femoral surface. And inside we told you that pelvic girdle actually forms a basin-like structure that is called pelvis. Okay, so this surface we call it as a pelvic surface. Okay. So generally the abdominal viscera, abdominal muscles, etc., they are in relation to the pelvic surface. So, and the inferior extremity muscles, they are generally related to the outer or femoral surface. Or there is, behind, this is also called the gluteal surface. This is the back of the inferior extremity, we call the gluteal region. There are so many muscles, which is gluteus maximus, gluteus minimus. So they are attached, they are related to this area. So this is also called the gluteal surface. And some muscles are originating and going down and inserted either in the femur or going beyond the knee joint. Okay, they are the femoral surfaces. Now, above the ilium, what we can see, what now I want to impress on your mind about the anatomical position, how we can hold it in anatomical position. Now, this is one spine we can see, spine-like structure. This is called anterior superior iliac spine. Again, we are getting here with the anterior inferior iliac spine. Similarly, posteriorly, we are getting one spine, that is posterior superior iliac spine, posterior inferior iliac spine. And so, we can find some borders which connects the anterior superior iliac spine and anterior inferior iliac spine. This is called the anterior border. Similarly, posteriorly, posterior superior iliac spine, posterior inferior iliac spine, this is the posterior border. Okay. And this is the superior border or the iliac crest. Okay. Now, if we go down, we can find there is a junction, this is an elevation which is called, any elevation is called eminence. We can find a bony elevation which is called eminence. In between ilium and pubis. So this eminence should be called as a iliopubic eminence. And more in front, we are getting one border and ultimately which ends in another tubercle. It is present within the pubic bone, which is called the pubic tubercle. From there, one linear structure we can see. This is called the pubic crest. Why I am uttering so many terms? Because the anatomical position is this anterior superior iliac spine and pubic tubercle should lie on a same vertical plane. Same vertical plane. They should lie on same vertical plane. Like if we put it here, then that will be the exact. This is the vertical plane. So this is the anatomical position. Okay. And in anatomical position, there are other points also. Then we can also find this bone. We can find this is the sacrum. Now, this pubic tubercle and the anterior superior iliac spine, they are in same vertical plane. There is one point. Another point is that there is an articulation between the sacrum and the ilium, sacroiliac joint, and we can find this one also. So this is the midline. So this one or the posterior superior iliac spine is how much away from the midline? It is around 5 5 centimeters. That means 2 inches from the midline. So, অবশ্যই বলবো কেন এটা কেন বাড়িবারে বলবো যতক্ষণ না বলে যাবে ততক্ষণ বলবো we can find this is the posterior superior index line and this is we can find this is the articulation between the sacrum and ilium. Okay. So this point we know this is the midpoint. So midpoint is how much away? 5 centimeters away from the posterior superior iliac side. Or in other words, from the midline, if we go laterally about 5 centimeters, we can find the posterior superior iliac spine. This posterior superior iliac spine, how we can identify in the living body? In a living body, we can find a dimple in the back that corresponds the posterior superior iliac spine. So this is the posterior superior iliac spine. কি গো বাবু হাসি হাসি করা দাঁড়াতে কাটু করা ভিডিও ভালো লাগে so this is the नाम क्या होगा हेलो रेविंग तो मिस्टी शॉप वाली
खूब स्वीट
so this is the posterior superior iliac spine we can find in a living body by a dimple in the back. So this is the iliac crest. Okay. And this iliac crest we can find one curvature. We can find if you see one curvature, one convex curvature is outside. Then we can if we trace we can find a concave curvature behind. So junction between the convex and concave curvature, this is the anterior segment and this is the posterior segment. So this is the iliac crest, aiming the or upper border of the ilium, which is called the iliac crest. Okay, this iliac crest is divided into ventral segment and the dorsal segment, and where there is a change of curvature. There is curvature to that. Yes. Convex, then concave. So junction between this is the ventral and dorsal segment of the iliac crest. Actually, the flat muscles of the abdomen, like external oblique, internal oblique, transversus abdominis, which forms the anterior abdominal wall. Actually, they are either taking origin or insertion over this crest. Okay. Absolutely outside there is one fascia. Actually, the fascia, what is meant by the fascia? It is a membrane-like structure. It is a connective tissue membrane-like. যখনই তোমরা দেখেছো এখানে মাংস ঘোরানো থাকে এটা হোয়াইট একটা মেমব্রেন দিয়ে so that is the fascia. There is a superficial fascia and there is a deep fascia. Deep fascia is actually covering the muscle and superficial fascia means just if you cut the skin then just below the skin we can find a superficial fascia. And below that we get a deep fascia. And deep fascia of the body in different place, it has got different names. In leg, in the thigh, it is called the fascia lata. fascia lata. Actually, it covers all the muscles and maintains a smooth contour of the muscles of the thigh. This is called the fascia lata. And fascia lata is attached above over the iliac crest, the outer border of the iliac crest. And within the fascia lata, there is one muscle which actually is inserted over the fascia lata, okay, which keeps the fascia lata tight, okay, which is called tensor fascia lata. Tensor fascia lata, which is also attached here. So fascia lata and tensor fascia lata, actually they are attached in the outer lip of the ventral segment of the iliac crest. সহজ করে বলছি, আরো মানে অনেক আরো স্পেসিফাই করা উচিত করব, তোমাদের পক্ষে আরো কঠিন হবে।
টেনসর ফ্যাসিয়া ওটা কি মাসল?
মাসল,
এটা মাসল, কিন্তু ফ্যাসিয়াল আর
ফাসা যে ফাসা ল্যাটা অ্যাকচুয়ালি মানে নিচ থেকে আসছে, then it splits to accommodate that muscle. Okay, মানে ভি শেপের হয়ে ঢুকে পড়ছে, মানে এরকম। বাকি আমি একসাথে নিয়ে নিয়েছি, মানে তো মুশকিল হবে। আরেকদিন আসছে। This is the fascia lata, that attachment, and in between the two layers of the fascia lata, because fascia lata splits up into two layers, and within the fascia lata we will get
টেনসর ফ্যাসিয়া।
So this is tensor fascia lata. Lata মাসটা কি? ঠিক আছে? Lata means in Latin হয়তো thigh. Okay. Fascia lata. Similarly, in our leg, there is another deep fascia which covers the muscles. Okay, that is called fascia cruris. Fascia cruris. Crura means part, limb.
So does this bone have any special quality that differentiates it from other bones because it is bearing the weight of the body?
True. Actually, the name of the fascia is given fascia cruris. The deep fascia, the same fascia, deep fascia, when it is crosses the knee joint, we call it as the fascia cruris.
So I'm asking, does this bone have any special quality that differentiates from other bone because it is bearing the full weight of the body? So
Yes, because definitely, because it is bearing the weight. Okay, so it is very much thickened in some place. You can see this line is very thickened. Actually, the weight is going through this area. So this is very specialty is that the bone here you can see this is very thin here, but it is very thick here because it has to bear the weight. This is the most characteristic feature. Actually, we were actually quadrupeds. Then we are gradually due to our evolution, we have become the biped. So there are some evolutionary changes beyond the scope of your syllabus. But the next question, yes, this is the area which bears the weight. So the bone is somewhere, and when and it is thickened here, it is also thickened here. Because when we sit, the weight goes through this area. When we stand, the weight okay. During these are two areas where the force is passing. Okay. So you can appreciate bone is very thick around this area. Because when we are sitting over this area. Okay. Ischial tuberosity. When you are sitting so, yes, this is the specialty of the bone. Specialization. Because it is very thick here in these two areas. Even that is also beyond your focus syllabus. Because within 1 year, you can assimilate so many things. You have to know the gross features and attachments. Very interesting. Why this is thickened here? After both the outer range. Very good. Don't know the
Jubair. Kolkata. Central Kolkata entirely.
how many intelligent are you?
5,000 offices.
White House, on the second floor. Namaskar. কত কাছাকাছি যাবেন? So now, so outside there is a partial layer, and then in between the two layers we are getting the tensor partial layer. And we can find a bony projection here. The bony prominence, which is called tubercle. So this is called, it is present in the iliac bone, so we will call it as a iliac tubercle. So anterior superior iliac spine, iliac tubercle. So this tensor fascia lata, his attachment is from this from the behind of the anterior superior iliac spine up to the iliac tubercle, and that muscle goes down. And it is inserted over the fascia lata. What is the action? When it contracts, it becomes very tense. And this tensor fascia lata actually it forms a thickened, which is called the iliotibial tract. From here, it forms the iliotibial tract. Very hard. If you can feel, you can find there is a iliotibial tract which passes down and ultimately is attached at the oval impression over the lateral side of the tibia, which is called iliotibial tract, which helps us to stand. যদি iliotibial tract না থাকতো, এই the tensor fascia lata is thickened here, which is called iliotibial tract. From ilium to tibia. So this is iliotibial tract. পরীক্ষার জন্য খুব ইম্পর্টেন্ট। iliotibial tract. Projection to এখান থেকে যাবে। Oh, one projection. This is tubercle. This is the this is the anterior superior spine, and if you go behind, around 5 centimeter behind, we can find this tubercle. This tubercle means any bony projection, which is called there. This is the iliac tubercle. There is another thing. There is the iliac tuberosity. Later on, no. Well, one is iliac tubercle and another is iliac tuberosity. When the elevation is widespread area, on a widespread area, this is a tuberosity. It is the iliac tuberosity. This is the iliac tuberosity and this is the iliac tubercle. This is the iliac tuberosity. Okay, because this is a bony projection, but it is a widespread. This is the iliac tuberosity, and this is the iliac tuberosity. Hello. Then we will get an attachment of one muscle, flat muscle, that is the insertion of one muscle, that is the external oblique muscle. Then we will get the insertion of the origin of the internal oblique muscle, and most inside there is another flat muscle which is called transversus abdominis, which is attached at the inner lip. So I am not concerned with the abdominal muscles now. Okay, leave it. In the dorsal segment, we are getting another, we can see the curvature, the concave laterally, and it is also divided into one part is, there is a bony elevation which divides one part, one lateral part and one medial part. Okay. In the lateral part, we will get the attachment of a very important muscle, that is the gluteus maximus, which forms the prominence of the back of the buttock region. Gluteus maximus. There are three muscles. Superficially, from superficial, and most superficially is the gluteus maximus. In the deeper plane, we are getting the gluteus medius and minimus. So gluteus maximus muscle is taking origin from the outer sloping area of the dorsal segment of the iliac crest. It's a dorsal segment though. This is the ventral segment. So this is the area for the gluteus maximus muscle. Muscle ta kothay ache? Back and where it is inserted? The 3/4 fiber is inserted over the iliotibial tract. এই মাসলটা উপর টেনে রাখে। Not only tensor fascia lata, but also this one. And 3/4 fiber are attached to this iliotibial tract. And 1/4 fiber is attached to the tuberosity, which is called gluteal tuberosity. তার মানে কি করে হলো? From here the muscle is taking origin, and only 1/4 fiber are attached here. Okay. And rest 3/4 fiber over the iliotibial tract. হয়েছে হজম হচ্ছে তো মানে পেট ভরে নিচ্ছে মানে একের সময় গ্যাস্ট্রোইসোফেজিয়াল রিফ্লাক্স ডিসঅর্ডার তারপরে করে স্যার একটু পরে আসছে আমি একটু পরে যাচ্ছি লাস্টে কি হবে অনেকে আবার জুতো দিয়ে আওয়াজ শুরু করে এখনো জল খাবার মতন অবস্থায় আছে জল খাচ্ছ মানে এখন আরে যতটা পারো খেয়ে নাও আমি পড়ার কথা বলছি কিন্তু there are some lines we can draw on this bone.
से चौक दिया उधर दिया
দেখো ব্ল্যাক বোর্ড টাইপ করে তৈরি হয়নি আমার পছন্দ ছিল ব্ল্যাক বোর্ড এটা দিয়ে দিল হোয়াইট বোর্ড তো যাই হোক ছেড়ে দাও
लेक्चर ছেড়ে
from here we can draw one line which will go upwards and will touch the iliac crest. How much away? 5 centimeter in front of the posterior superior iliac spine. So one line we can draw here. This line is called posterior gluteal line. This is the posterior gluteal line. Posterior gluteal নেই কিছু আছে পড়া না তারপর আবার অ্যালিটা লিখতে সবকিছু ভুলে যাবে from this area is called the greater sciatic notch. We can find one notch here. This is the name. The name is greater sciatic notch. Similarly, another notch is there, lesser importance level. From here, another line will come. We can draw. From the same the greater sciatic notch, we can draw another line. It will touch the iliac tubercle just in front. It is continued up to just in front of the iliac tubercle. And another line we can draw from here up to the. This is the superior superior iliac spine. This is the anterior. এখানেও আছে anterior superior iliac spine, anterior inferior iliac spine, posterior superior iliac spine, posterior inferior iliac spine. So this is another line. So this is the posterior gluteal line. This is the anterior gluteal line, and this is the inferior gluteal line. পেছনে আছে posterior, এটা আছে সামনে, inferior মানে আমাদের তো যদি anatomical পজিশনটা ধরি তাহলেই বুঝতে পারবে। হ্যাঁ, inferior, anterior, posterior, inferior. Now what is the importance? এত আমরা বলছি কেন? ওখান থেকে মাসেলের gluteus maximus muscle, not only it takes origin from this area, it also takes origin from the area behind the posterior gluteal line. এত বড় মাসল কি এইটুকু জায়গা থেকে অরিজিন করলে হবে? কাটের দিকে থাকতে হবে। তাহলে তুমি একবার টানলে, যে বন্ধু টানলো তোমার আপনি চলতে পারবে না, বসে উঠতে পারবে না, সমস্যা So, gluteus maximus takes origin from the area behind the posterior gluteus line. Posterior gluteus line. মানে এটা কি আমরা ঢুকে যেত লাইনগুলো প্রথম থেকে লাইনগুলো এটা হল this is the greater sciatic notch. This is the sciatic notch because sciatic notch actually comes out through this area. Greater sciatic notch, lesser sciatic notch. We can find from the greater sciatic notch one line we can draw upward. It will touch the iliac crest or superior border of the ilium. How much? Away 5 centimeter in front of the posterior superior iliac spine. Here. Another line we can draw from the same point from the greater sciatic notch. It will go up and will reach up to the iliac tubercle. This is the anterior gluteal line. The gluteal line, gluteus maximus is done. Gluteal surface. And this is the inferior gluteal line from the same source to the anterior inferior iliac spine. বুঝতে পারছো বাবু? তুমি তো বহু দূরে আমার থেকে, তুমি হ্যাঁ হ্যাঁ হ্যাঁ, মাথা ঘোরালে হবে না, তুমি বহু দূর।
বলছে আমরা দেখতে পাচ্ছি।
অ্যাকচুয়ালি এই ক্লাসটা মানে ছোট হয়, কিন্তু বা এখানে যদি একটা ফাঁকা জায়গা থাকতো তাহলে আমি যেতে পারতাম, সব তো ফাঁকা জায়গাটা রাখিনি। So there are 3 lines. Now this area behind the posterior gluteal line is the area for the attachment of the gluteus maximus, and in between anterior and posterior, this is for the attachment of gluteus medius. Maximus, this is for medius, and between inferior and anterior, that is a minimus. Gluteus maximus is in a superficial plane, and gluteus medius and minimus in a deeper plane and the same plane. বডিতে দেখলে তখন বুঝতে পারবে, কিন্তু এখন তখন বডিতে আবার বোনস দেখতে পারবে না, তাই না? তখন আবার বোনস দেখতে পারবে না, তখন if mind doesn't know, eyes cannot see, তাই না? আবার এটা বলতে পারো যে রবীন্দ্রনাথের গান ছিল যে চোখের আলোয় দেখেছিলাম চোখের বাহিরে, চোখের আলোয় তুমি এখন দেখে নাও, এবার যখন ওখানে কিন্তু চোখের বাহিরে যখন দেখতে পারবে না তখন কল্পনা করবে যে yes, this is the area for the gluteus maximus, this is for the মাসেল ওয়ালা কোন সেট পাশের রুমে আছে, ওটা দিয়ে read and repeat এর কোনো ইয়ে নেই, সবসময় ভাবতে হবে এখানে মাসেলটা কোথায়, this takes origin from this area and ultimately inserted over the femur. So what is the action? This is the flexion, extension, the extension of the extension of the hip. ঠিক আছে, আরো আছে, তো at least you should know this is the extension of the hip, the gluteus maximus, gluteus medius and gluteus minimus. Okay, this is greater sciatic notch, এত বড়, this is the lesser sciatic notch, এইখানে স্পাইন মতো থাকে, ischial spine, স্পাইন মতো, ঠিক আছে? পরে তো নাও pelvic surface বলার ইচ্ছে নেই। আচ্ছা, next is the very important point for the lower extremity, this is the anterior superior iliac spine. আশিস
দৌড়াচ্ছে
মেসির মতন কিন্তু
এটা বইতে লেখা ছিল না।
আশিসটা বলেছে anterior superior iliac spine, in short form it is called ASIS. So what is the importance? Actually the superficial most flat muscle is the external oblique. This external oblique muscle is thickened here to form a ligament which is called the inguinal ligament. The anterior wall of the lower abdomen is called the inguinal region, where we get a ligament, very important ligament, which this is called inguinal ligament, because when the same blood vessel, which is called external iliac, when crosses this ligament, that passes below this ligament, it becomes the femoral artery. So this ligament, most important ligament is the inguinal ligament. It is attached at the anterior superior iliac spine. And the other end, there's a lateral end is attached here, and medial end is attached in this tubercle, which is the pubic tubercle. Okay, so this is the ligament. Okay, ligament এরকম আছে. Okay, this is nothing but the thickening of the anterior abdominal wall. There is a superficial flat muscle, superficial most flat muscle, that is the external oblique. Deep to that, internal oblique, deep to that, transversus abdominis, deep to that, fossa transversalis, deep to that, parietal peritoneum, deep to that, abdominal. বাম্পার আসছে So this is the anterior superior iliac spine, and another ligament, another very important muscle of the lower extremity, that is called Taylor's muscle. Taylor's muscle, Taylor না দেখবে যখন Taylor দর্জি Taylor মধ্যে যখন জলটা ফুটে মানে জামা কাপড় সাজতে যারা তুমি তো বলো এমনি পরে এত সুন্দর একটা জ্যাকেট So Taylor, what is the posture? This is the flexion of the hip joint and flexion of the knee joint. And this muscle is taking origin from the anterior superior iliac spine and is inserted over the medial aspect of the tibia. So when this muscle contracts, so there is flexion of this hip joint and flexion of the knee joint. Okay, continue. So this posture is, we can find this posture in Taylor's. Right now, তারা দু পায়ে তো মেশিন চালাচ্ছে. So that is this muscle is called the sartorius muscle. Sartorius muscle, which takes origin from here, and it is inserted. Where it is inserted? Is the medial surface, the upper medial surface of the tibia. The medial surface is very much subcutaneous. You can feel the medial surface in your medial surface of the tibia. We can feel it's subcutaneous. The chamar tole দেখো বলতে আছে. So in the upper part we get the attachment of three muscles. One of them is the sartorius muscle. Other two are comparable. The hamstring muscles. This muscle is called, this group of muscle is called the quadriceps muscle. যারা একটু তোলাতুলি করো জিম জাম্প যারা করে তারাই হচ্ছে এসব জানে, তারা জানে। This is the hamstring, this is the quadriceps muscle, this is the hamstring muscle. Okay, this is the leg muscle. So all the hamstring muscles, there are hemitendinosus and tendinosus. একটু পরে এটা পরামর্শ নেই, তবে আপনি ইলিয়ামটাই হয়। তারা আবার বলেছিল বেশি ইন্ট্রোডাকশন করে ছেড়ে দিই, হালকা নেক্সট another important, there's one ligament is attached here and one muscle is the sartorius muscle. This process like this. Okay, sartorius muscle is originally anterior superior spine and insertion also to the tibia. This is the sartorius muscle or Taylor's muscle, which causes the flexion of the hip and flexion of the knee. Now anterior inferior iliac spine. Here we will get an attachment of a similar one muscle and one ligament. One ligament from, this is the strongest ligament of the body, this is the iliofemoral ligament from ilium to femur. So stem of the stem, this is Y-shaped ligament, stem is attached here. আর ওয়াইটা পরে নিচের দিকে নামছে ভুলে যাব the stem of the iliofemoral ligament is attached in the anteroinferior iliac spine and there is a notch we can find in between and there should be a spine and anterior inferior iliac spine. One nerve comes out through this area, just below the anterior superior spine, which nerve is coming from the dorsal abdomen, passing over this. There is a one muscle which is called iliacus. Then it comes out and it is distributed over the lateral aspect of the superficial or skin of the thigh. The name is lateral femoral cutaneous nerve. Laterally distributed over the femoral region, so femoral, lateral femoral, and it supplies the skin, which is called the, so it is a cutaneous, lateral femoral cutaneous nerve. Good value? L1, L2. L2, L3. Lumbar Texas okay, this is the nothing but good value is L2, L3. Okay. L1, L2 is the genitofemoral nerve. Forever. খুব সহজ বুঝাবো সহজ so that is this nerve, this was the lateral femoral cutaneous nerve and what is the importance is that in diabetes or in myxedema this nerve is affected ধরো এখানে প্রচন্ড তুমি এখানে থাইটা ফুলে গেছে এখানে fluid accumulation or peripheral neuropathy in diabetes, this nerve is affected and there is typical tingling sensation, sometimes which is called paresthesia. The sensation is exaggerated in some peculiar manner. This is called meralgia paresthetica. আর পাচ্ছি না মিরালজিয়া প্যারাস্থিটিকা বাবাকে বলবো আমি কালকে আমি হলো রইল ঝোলা চললো বোলা মিরালজিয়া প্যারাস্থিটিকা এসব ওরা একমন না স্যার বাড়িতে আমি বাড়িতে বিদ্রোহ করবো আমাকে কেন আমাকে ডাক্তারি করতে দিয়েছি কি তোমার হয়েছিল আমার ছেলেকে ডাক্তার বানাবো মেয়েকে ডাক্তার বানাবে মিরালজিয়া প্যারাস্থিটিকা মনে থাকে বাবা এসব কি বকতে পারে আমাকে মনে রাখতে হবে মিরালজিয়া প্যারাস্থিটিকা নালে কি বলবে ডাক্তার বাবু তেতো নিয়ে ঘুরবে So this is the neuropathy of the lateral femoral cutaneous nerve. This is called neuralgia paresthetica. আমার নাম ডক্টর সিবি নারায়ণ চৌধুরী। তুমি রামপুরহাটের লোকেদের থেকে শুনেছো বোধ হয়। কলকাতাতে ন্যাশনাল হসপিটাল। হ্যাঁ আরো মজা করে নানান রকম ব্যাপার আছে মজা আছে so this is about some now this is the area. This is called the iliac fossa. Fossa means we can keep something like locker. This is the iliac fossa. This is present in the iliac bone. Okay, actually these three bones, actually they are divided in the, actually they remain separated during embryonic life, also in the child life. Okay, they are divided like a Y-shaped cartilage. কিরকম? This is called triradiate cartilage. We can find inside the acetabulum in a young bone. Okay, in a young bone, we can find a Y-shaped cartilage, or it is called the triradiate cartilage within the acetabulum or acetabular cavity, and this part actually belongs to ileum. This belongs to pubis, and this belongs to ischium. Okay, and they remain separated. There are three pieces of bone: ileum, ischium, and pubis. They remain separated by a cartilage. It's a triradiate cartilage. Okay, epiphyseal cartilage. উপরে একটা খন্ড, নিচে একটা খন্ড, মাঝখানে লম্বা একটা খন্ড, in between ছোট ছোট খন্ড। এটা diabetes, epiphysis, and epiphyseal cartilage. So this is also epiphyseal cartilage, and they get united at around 12 years of age. আবার একটা ইনফরমেশন মনে রাখো, ঠিক আছে? মনে রাখো। So this is the ileum. So this is the iliac fossa, and this is for a muscle, attachment of one muscle. This is called iliacus muscle, which it is taking origin from here and inserted over the lesser trochanter. Okay, in the base of the lesser trochanter, they say iliacus. And there is another muscle which is coming out from the posterior abdominal wall, and straight where it is coming out. Along with these two muscles, they are inserted over the lesser trochanter. Okay. So this is iliacus, and another muscle is the psoas major muscle. They both actually forms a bulk, single bulk of muscle, and ultimately attached to the lesser trochanter. Okay. So, and this muscle, the other muscle, the fleshy muscle, that is called psoas major muscle. This is taking origin from the bodies of the lumbar vertebra. পেছনে শুয়ে আছো, এই যে উঠছো, শোয়া অবস্থা থেকে এই শ্বাস মেজর কন্ট্রাক্ট করছে বলে উঠতে পারছো। শুয়েছিলেন, শ্বাস মেজরই বেসিক্যালি, and when they are both the iliacus and psoas major, they are contracting. Flexion. So it flexes the hip and it helps to forward movement of the trunk from recumbent position to sitting position.
উঠতেই হতো না জীবনে না
উঠতেই হতো না। যদি মাসেলটা না থাকতো, তুমি না থাকলে এত সমস্যা হতো না। So this is the আর পেটের যদি ওই 1.5 লিটারের স্টমাকটা না থাকতো তাহলে সবই ঠিক করলো নেচার, কিন্তু এটাই গন্ডগোল করে দিয়েছে, তাই না? সবচেয়ে বড়টা যদি স্টমাকটা না দিত তাহলে বেঁচে যেত, তাতে এত মারামারি হতো না, এত হিংসাহিংসি হতো না। There are so many causes, not only one muscle, there are so many other points. The ligament, when the ligament is affected, it can have when the muscle is affected, it can have and in case of appendicitis, what happened? This muscle is passing through this area. An appendix is here. On the right iliac, this is the iliac fossa, and this is a right iliac fossa, and within the right iliac fossa we get the cecum and the appendix. So appendix is in relation to the psoas major muscle. Okay. So when there is a contraction of the psoas major muscle, there will be pain. পেশেন্টটাকে শুইয়ে দিয়ে পাটা ধরে এরকম আমরা টানি, মানে মাসলটাকে স্ট্রেচ করে চেঁচিয়ে ওঠে, মানে সোয়াস টেস্ট। That is called the সোয়াস টেস্ট of appendicitis, acute appendicitis. শুধু এর উপর টিপে টিপে দেখে দিলে হবে না, এসব কিছু পরীক্ষা করতে হবে, সোয়াস টেস্ট কনফার্ম করা যাবে। ওকে, তাহলে তুমি ওটা টেকনিশিয়ান ওটা টেকনিশিয়ান একটু দেখে দেখে বলে দিলে এই তো এটা তোমাকে বলতে হবে what is McBurney's point, what is McBurney's tenderness, একটু জানতে হবে। ঠিক আছে, চলো।